It's not pretty. Good morning, everybody, and thank you so much for joining us this morning. Welcome to everybody, and in particular, those of you who braved the APEC-associated traffic this morning to make it here in person, as well as those of you enjoy online. Welcome. We're delighted to have you here. I'm Sue Lynch. I have the pleasure of serving as the director of the Benioff Center for Microbiome Medicine. And before we get started today, I'm just going to kick off with just a little overview of our center and what we've built over the last 4 years since we launched the center. And the original vision for this center was to build an inclusive, collaborative microbiome research hub that really democratize the field. There's several of us that this is our bread and butter. This is what we've always done. And we want to facilitate that research, but we also want to engage a number of investigators who maybe are on the fringes or really like the field or really just want to become engaged. And to do this, we've built out a number of major programs in the center, an education program that includes summer internships, workshops that are all made available and open online to investigators, as well as our monthly Ignite seminar series that I encourage you to join. Again, we firmly believe in open science and want you to engage and to understand and learn about what's going on in terms of the research that's happening at the center here on campus. We're also firm supporters of diversity, equity, and inclusion, and are incredibly proud of all of our efforts in this area. Not least our summer internship program with Mayor London Breed's Opportunities for All program, as well as more recent efforts led by our colleagues at UC Berkeley, a National Science Foundation funded effort to diversify the microbiome field. And that's in collaboration with amazing colleagues at San Francisco State University and Cal State East Bay. And I'm incredibly proud to have several of the first cohort of these RAMP scholars here at UCSF embedded in microbiome labs. It's just been an incredible program, and we're really proud to be part of it. In terms of research, it astounds me every time I see the breadth of work that's going on across campus. We've over 120 faculty and investigators as part of the center. And the research that goes on spans all four of our schools: medicine, nursing, pharmacy, and dentistry. It's really just astounding to see the work that's going on, and that's facilitated by a number of infrastructural efforts and facilities that we've built on campus. The gift that was provided by Mark and Lynn Benioff has allowed us to build a brand new gnotobiotic animal facility led by Jesse Turnbaugh, which will open in early next year. We're also in the midst of renovating space on campus to form a central home for the center in one of the research towers here on campus. But I think for anybody who saw the large hole in the ground and the hillside on the west end of campus, we're really, really excited about the new Parnassus Research and Administrative Building that will open in the next couple of years. And we've been granted a large amount of space in that brand new research building for the Microbiome Center. And we're just incredibly excited to call that our ultimate central home. And these efforts and the provision of space and infrastructure has allowed us to attract several new faculty members into the center as well. Exciting hires, including Chris Hernandez and Kelsey Collins, as well as Sumaya Zlitny, who will be joining us from Stanford early next year. All have exciting microbiome research programs and have become really integral members of the community here on campus. And ultimately, it's all about the research, right? It's about fostering collaborative, inclusive research. That's what we're after here on campus. And I'm just thrilled for the lineup of amazing broader Bay Area and beyond research that we're going to hear about today. I am truly really excited for the lineup of speakers that we have this morning. We've got sessions that will be moderated by members of the Microbiome Center's Executive Committee and by two of our trainee ambassadors, McDad and Margot. And before we get started, we'll perhaps go through a few housekeeping items. If you're virtually joining us and you have a question, please use the Q&A function in your Zoom dashboard, and our moderator will address your question here in the room. If you're here in person, raise your hand. We'll get a mic to you, and you can ask your burning question that you have. I particularly want to thank our guest speakers for today. I know several of you have traveled. Some have traveled from outside of the Bay Area. I'm really, truly, I know everybody is stretched and everybody has a lot on their plate, and I'm truly grateful for you for your presence here today and sharing your newest research with us. I really am incredibly grateful also to Nancy Cook, Kristen Dolan, Peter Turnbaugh for their efforts to really bring this symposium to us. You know, we start planning it as soon as we finish today. We pretty much start planning for the next year. It's a lot of work, it's a lot of effort, and I'm just really grateful for what we get to do here today, what we get to witness and experience. And it's all through, you know, Nancy, Kristen, and Peter's efforts. And I also want to call out Sarah Mortley, who was part of our administrative team until the summer of this year when she moved back to Melbourne to build another centre there and be part of that initiative. Sarah really helped with the initial kind of organization of this year's symposium before she left us. Also want to thank Educational Tech Services, in particular Dorothy Wang, who's up at the back and has just been instrumental in helping this run smoothly today. Thank you, Dorothy. We're really grateful for your efforts also. And just a reminder that the agenda for today can be viewed the QR code that was circling on the slides a little earlier on. And so with that, Micdad is our first moderator. Micdad is one of our trainee ambassadors. These, you know, I really wanted to know, what does our trainee community need? And we don't always have enough communication and contact, particularly during the pandemic, to understand the needs of our trainees. So we set up these trainee ambassador positions to serve as a conduit between you guys, the trainees, and the executive committee. And the trainee ambassador sits at the table with the executive committee and brings, you know, requests, requirements from the trainee community to us, so we can really serve needs of those on campus. And Micdad has served that role for a number of years now and has just been exceptional in this role. He's rolled out a number of different initiatives based on the needs of our trainee community. And for that, I'm also very grateful, Micdad. And so with that, the floor is yours. Moderate away. Thanks so much, everybody.
Good morning, everyone. So I'm here to moderate the first session. Our first speaker is Simone Roux. I hope I pronounced the name right. Simone leads the Viral Genomics Group at the Department of Energy Joint Genome Institute at Lawrence Berkeley National Labs. He received his PhD from France, where he was studying bioinformatics, ecology, and evolutionary biology. Simone's research interests revolve around viral diversity in the environment and impacts of virus-host interactions on individual microbes and on ecosystems. His work includes the development of bioinformatics tools and viral data sets, the analysis and meta-analysis of viral genomes assembled and metagenomes to investigate virus-host interactions in nature. And today he'll be talking to us about connecting viruses to hosts, the missing piece in viral ecogenomics.
Let's see. Here we go. Thank you. Thank you so much, and for the introduction, and thank you again for the invitation. It's a pleasure to be here and to open this session this morning. Like what just said, I'm Simon Roux. I'm lead viral genomics. We sit in the metagenome program at the JGI. The Joint Genome Institute, for anyone not familiar with it, is a DOE user facility. Essentially, we provide sequencing and nowadays kind of any kind of omics that you would like to researcher in the Department of Energy sphere and kind of mission. So I won't have time to talk about this too much, but if you are curious about what is a user facility and what is the national lab, please come and see me afterwards. I will be here all day. And because I have too many slides, I wanted to make sure I go through the thank you slide. As you can imagine, these are like big team projects. 3 people I want to highlight in particular here that I will showcase the work of. Clément Coquelet, a postdoc in my group at JGI, Antonio Camargo, a scientist in the microbiome data science group at JGI as well, and then Yuna Wang, a PhD student in the lab of Peter Girgis in Harvard. But anyway, it's we are doing big team science essentially at the national lab. That's what we're supposed to do, and that's what is represented in this slide. Okay, and just trying to go through this in the next 20 minutes. Why should you even care about viruses? A little technical part, I promise to keep it short, on like what are the newest techniques to link viruses that we know only from sequences to the host or putative hosts. And then the last part about like what can we learn from this and what should we be careful with. I usually have a slide before that say microbes are super important. I removed it here because I assume everyone knows and loves microbes there. And everyone knows that most life on Earth is microbial. What you may or may not have kind of made the connection to is like on the flip side, on the virus world, it's also true. If most of the life is microbial, most of the viruses are viruses of microbes. And we like to showcase it that way. For anyone who has never seen this kind of really pretty microscopy observation, this is a drop of seawater in this case, stained with cyber green, which reveals DNA. You have big dots, I assume. Where is my pointer? Can I show my pointer somewhere? No. Never mind. How interesting. Big dots are cells, small dots behind, the small regular dots are all virus particles. And you can count, but basically it's a reminder, and I have some numbers up there. The reminder is microbes and microbiomes live in a constant state of being infected by viruses, always, all the time, and that's just ongoing in any microbiome that we have looked at so far. So what do the viruses do? A few things. The first thing that is intuitive is viruses kill their host. When you think of this in a microbiome context, what this means is you have to remember that viruses tend to have, at least viruses of microbes, tend to have pretty narrow host range. It kills very specific microbes. And so in the context of a community, what this means is this will shape, or at least contribute to shape the microbiome composition. Because based on the activity of some viruses, you will have more or less of this specific microbe. But that's not all they do. Actually, I forgot about this. Yes. Because this is a microbiome center, also, I wanted to kind of bring this in. We are trying, and we as a field, of course, DOE is not directly also starting to look into this a bit, but like we as a field are trying to use the specificity of phages, viruses of microbes, to try to use it to, to our advantage, basically, trying to target very specific bacteria, archaea, even small eukaryotes in microbiomes to try to either remove them or enhance them, at least try to manipulate microbiome in a broad sense. So in the health context, it's phage therapy, but you can think of this way more broadly. If you want to change the composition of a microbiome, usually you can just look at the viruses and you will find the toolkit you need to do this kind of microbiome manipulation. But again, I want to kind of emphasize that this is not all what viruses do. And although they do kill the host, most of them, what they do during the infection is also very important from a microbial ecology standpoint. So that's what's represented here. Basically, what happens during the infection is important in the sense that an infected cell behaves differently from a non-infected cell. And as we saw from the previous slide, there are a lot of viruses out there, so a lot of infected cells in nature, and infections can be long. Like, in this bacteria, which is in this case, the cyanobacteria in the ocean, infections can last 11 hours. It's a very long process. And what happens during the infection is the typical metabolism of the cell is being rewired in a sense and kind of reprogrammed by the virus to its benefits. In this case, and usually it's the case, it's just diverting every resource towards building more nucleotides and amino acids, because that's the building blocks that the virus needs. But in the grand scheme of things, that means a profound change in the metabolic input and output of the cell. And if you have enough infected cells, that's, again, something that will be reflected at the microbiome level. So something to keep in mind, again, infected cells behave differently from uninfected cells. And that's true for like these very active, viruses actively replicating and producing new viruses. That's also true for viruses that we call sometimes dormant, prophages, lysogenic viruses, all of these kind of revolve around the same concept, which are viruses that are not actively producing new viral particles. They may still express genes, and sometimes they're actually actively integrated into the host genome. They are not actively producing viral particles. They may still express a few genes from their own genome that will, again, change the behavior and change the phenotype of the host cell. The most striking examples are these prophages that encode toxins, and you know the list. These are some of the heavy hitters there. And these are all at least partially prophage encoded. I don't want to claim like all of them are, but basically, we have prophage encoding example of all of these toxins, which again means that some bacteria only becomes really toxic and a health problem if they are infecting themselves by a prophage that confers them this toxicity. So again, killing the cell, but also changing the cell during infection. So at this point, my pitch is we should all care about viruses if we care about microbiomes. They are super abundant, they are found everywhere. We know there are lots of viruses out there, lots of viral infection going on in every single microbiome, and we know they can have massive impact. So now comes the question, okay, if we care about viruses, what do we need to understand? That's pretty much what the field of, broadly speaking, viral ecogenomics is after. Which viruses where, who infects whom, and who is active when. So that's kind of what we need to get as a broad, picture to then understand better and characterize and model better how viruses impact microbiome processes. That's easy, right? Now comes the kind of difficult part. How do you even start to do this? Well, one thing is we don't have 16S. That's a given. We don't have a universal marker gene for viruses, so we can't do PCR, we can't do survey by barcoding, metagenetics, whatever you want to call it. We don't have this at our disposal. But what we can have nowadays is metagenomics. And that has really become one of the main ways, if not the main way, by which we explore at least viral diversity. It's not the only thing we do, but it's really the main kind of first approach when you are like, want to explore viruses in a new microbiome, or you want to start your investigation into viruses. Usually you would start with metagenomics. So that's kind of my simplified diagram here. You can start from microbial metagenomes, or you can also do some treatment to your sample to try to remove the cells and only look at the viruses. In any case, what you end up with is you extract DNA, you shotgun sequence, and then you assemble your genomes. That's become pretty standard at this point. It works extremely well with viruses because the virus genomes tend to be shorter and they tend to have less regions that are conserved across different viruses. We have less issue with like fragmented assembly, and nowadays we can very routinely assemble complete phage and virus genomes from metagenomics. That's become relatively straightforward at this point if you sequence deep enough. I have DNA here. Technically, it also works with RNA. And you can also do this for like bulk RNA sequencing, or you can also kind of try to enrich for virus particles and then only sequence the RNA if you know that there are RNA viruses in your sample. I won't talk about this today, but if you are curious about this, we just had this paper, which basically mined publicly available transcriptomes to show that we can recover a lot of RNA viruses, including new RNA phages and all that. So if you are an RNA virus person, check it out. Now, of course, once you have like the assembly, you get like a lot of contigs. Your very first step every time will be you identify which of these contigs are viral, and that gives you your kind of primary data object, which is this set of virus genomes. That will be the basis of everything you do downstream, because once you have all your virus genomes that you have predicted from your metagenomes, then you can do all the rest. You can look at the function that are encoded on this genome, look at the distribution of this genome, which viruses were kind of thing, try to classify it taxonomically. And of course, because you have only a piece of genome, what you want to know is, I know this is a virus. I know it's there, and there. What host does it infect? And so this host prediction part is really important, and it's a key piece of how we try to interpret and better understand viruses from these metagenomic approaches. And again, I want to emphasize, this is really the main way, especially because we don't have PCR approaches available, that we explore virus diversity in microbiomes at this point. So how much does this really translate into in terms of impact on databases? That's my favorite plot. This is how many virus genomes we have in NCBI from 2004 to almost today. I should probably update this at one point. These are all viruses, include like human viruses. I just like remove all the different copies of flu and COVID because otherwise this would be like very skewed. But otherwise, these are like all types of viruses in NCBI. You can see we kind of accumulate them more and more. We sequence more and more. That's all good. And now we have overlaid how many viruses we assembled from metagenomes in the same span. And you can see that basically it's starting in 2016. I think I have it, yeah. Starting in 2016, it really started to increase. And since then, it has been a super, super quick rise. We are at like 5 million right now, but we are preparing the next version of our database. And we should probably, cross, fingers crossed. I mean, we'll see. I'm hoping for 10 millions, we'll see. I don't know for sure. But basically, we keep increasing for sure because we see new metagenomes, which are deeper, like, more deeply sequenced. The assembly becomes better, so we get more and more virus genomes. That's how fast we can accumulate virus genomes from metagenome at this point. How do we find all of this? One thing I want to highlight, if you have a metagenome and you want to find your viruses, check out this new tool that Antonio Camargo just published. Again, it's from JGI, it's called Genomad. It's probably the latest and best thing in and brightest thing in virus sequence detection. The good news is Genomad has been developed as very agnostic to the type of virus you want to find. So it will work for eukaryotic viruses and viruses of microbes like phages, archaeal viruses, and everything. It works for DNA and RNA viruses. So, if you're into this kind of analysis, you should definitely check it out. I found it really, really improved compared to everything we had before, including my own tools. The other part, if you're curious about this data, these 5 millions, we have this as a big database. We call IMGVR. You have the URL here. You can see here that's kind of 5 million is what we call the high confidence virus. We actually have 10 million other sequences, which we call putative virus, because it's too short to be sure if this is virus or not. But the tool tells us it's probably a virus. You can definitely explore and play around with this caveat that we are not completely sure. These 5 millions, we are very reasonably sure that almost all of them are real viruses coming from about 20,000 metagenomes, so across a broad range of ecosystem, broad range of environments, and all this. Okay, so what do we have in IMGVR? And that will be my transition to why host prediction, basically. What we did is we gather these 5 million and we try to make them useful, right? We don't want to just dump 5 million sequences and say, okay, have fun. I mean, it's cool, but like, can we do a bit more to help you kind of select which of these 5 millions will be interesting for you? So we look at the genome quality and completeness. We look at, which viruses are found in which ecosystem. We do some taxonomic classification, and we try to do host predictions. The idea being, let's say you are someone very interested in a specific host. You could come in and say, okay, give me all the viruses linked to this host. That would be ideal, right? That's the current state of host prediction in IMGVR. So we have host prediction for about like 7 to 10% to the genus family level, and we have 80, 90% of the sequences for which we have no predicted host. We know there are viruses. We don't know which ones are in fact. And the database was built in 2022, so that's kind of state of the art for 2022. If you have ever done this kind of analysis on your own data, most likely you have seen a picture like this. It's not as bad for human microbiomes usually because we have better references. So, I guess good for you. If you're in soil, if you're in like extreme environments, if you're even in like freshwater lakes, you have seen something like this in terms of host prediction. Most of the recent tools still had like a lot of issues in 2022 to get enough, to get host prediction for most of your sequences. That brings me to my second point, which again, I think I hope I have illustrated clearly, like this host prediction is a key part of the pipeline, right? If you don't have a host predicted, you're kind of stuck at one point in what you can say or understand about your viruses. So that's why we really went after it and we said, okay, we need to build a new tool that is better than what we have. It's not a new idea. We have many, many tools that do host prediction out there. And of course, everyone wants this. So everyone has developed their own tool. So we had like, when we started in this project in 2021, at least 15 different tools that have been published in the last 2 years. All of them had limitation. Either sensitivity, meaning they work very well, but only for a few types of viruses, or they were, very not specific, meaning like they gave you a lot of predictions, and a lot of them were right, but a lot of them were wrong, and you couldn't tell which from which. Or reference bias. You have tools that are very sensitive and very specific on benchmarks, but they need a known phage or a known virus to make this prediction, which is great if you are working in an environment where you have a lot of references for. Again, not great if you're working in an environment where all the viruses are new. So what we did is kind of simple. It's like, well, we have all these tools. They seem to all work great and all have their flaws. What if we all mush them together, do some machine learning magic, and get one prediction from all of them? And maybe the machine can learn like, okay, if I have this and that, and then I can trust this one. And if I have this one, tell me the same thing, then that's also even more confidence. And hopefully we can get to this kind of, okay, not only I have a host prediction, but I know how confident I could be in this host prediction. And it sort of worked pretty well. That's a tool that we developed called IPOP, which is published right now. You can get it also on, all of your like nice platforms like Conda and everything. The reason why it works is, again, because we use all of these different tools, we can be better at distinguishing what are the real predictions from the erroneous predictions. And that's basically the benchmark. I don't want to go through it too much, but basically all the other tools are different colors. The IPOP is in blue, the biggest bar, of course, because I'm presenting this tool, so it's always the best one. Kidding aside, though, the thing that we really like is this part. So the x-axis here is basically a proxy for how close from a reference is your input virus. Is your virus like 60 to 100% identical to a non-reference, or is it 0 to 5% identical? So 0 to 5 is essentially it's a new virus. You have nothing that looks like it in a virus database. And of course, if you have something that looks like this in the reference database, host prediction works really, really well. And we still get a prediction for half of the ones that are completely new, which is really what I was after because I'm a microbiome ecologist at heart, and I want to find the new viruses and everything. But again, we want a tool that works across this x-axis here, and we're really happy with that. The other part is, of course, can we get, when we use it on metagenomes, do we get predictions, high confidence prediction for most sequences? And the response was yes, pretty much. You can see we broke it down here by ecosystem because some ecosystems are much easier to predict than other. But you see, if you have, viruses and like IPOP is specifically for phages, so I should switch to phages here. If you have phage sequences from the human-associated microbiomes, you basically get a prediction for almost 95% of your sequences, and a high-quality prediction is dark green for more than half of them. That's really good, again, because we have a lot of reference sequences from the phage and the host side. So we can do a lot of kind of matching, and we have a lot of temporary phages as well, which are easier to predict the host for. All the other ecosystems, not as good. Still, we do reach this 50% mark if we go to like the more like lower confidence, probably family level-ish host prediction, but we still get something, which is much better than we used to have. And we are hopeful that by building up the reference databases and by discovering more viruses and characterizing more microbes from the different environments, we will reach eventually this kind of state that we have for the human microbiome at this point. Okay, that was my technical part, what we did and why we think it's really important, and what is the kind of bright future ahead of us with the host prediction. Now comes the fun biology part. So again, what we're trying to do is, I told you we can get very easily at this point, I mean, pretty easily at this point, you can get a big genome catalog of viruses. But now you want to understand what are they doing and why. So that's kind of what Clément did in my group for this first project I want to share, this History of Watershed project. We are talking here about a model ecosystem that has been studied at Berkeley Lab for a long time. There are a lot of samples taken all across the watershed. There is like climate modeling, like large scale modeling, and then small scale and everything. So it's a really kind of nice study site. And especially what's happening is there's been some study about the microbes in the soil of this watershed, and also about like the nitrogen pulse when the snow melts. I should have mentioned this is in Colorado. So the beautiful picture is summer. Of course, winter is full of snow. And then it goes through a strong seasonal transition. And apparently you can see like the y-axis is like nitrogen that is measured in the soil or in the pore water, I think. And basically every year in like June, which is kind of snowmelt, like the end of snowmelt in Colorado, you see a big pulse of nitrogen released. And we know we suspect it's probably microbe related. But the question is, why do we have all this nitrogen released? And could it be maybe viruses involved in maybe killing some host and kind of recycling nutrients, because like that will release a lot of like nice nitrogen in the environment. So that's kind of what we were after in a way. More broadly, we wanted to understand, okay, which viruses are there and how do they change between under the snow, we have samples under the snow, all the way through the seasons to like September, which is when the plant die and the system kind of goes back more into like a dead-ish senescence kind of state. So what we used is we had metagenomes and metatranscriptomes across 4 dates, these kind of 4 seasons, 6 locations and to replicate to make sure we had like about different types of soils along this kind of watershed. So that ended up being like about 50 metagenomes and metatranscriptomes, and we have host prediction, again, using IPOP. I won't show all the data, but what I want to show is what you gain when you have a combination of like metagenomes, metatranscriptomes, so RNA sequencing, so virus catalogs that you can see which genes are expressed and then connect them to the host. What we gather all this, and we looked at this, that's what we came up with essentially. So this will be a complex figure, so I'll just like walk you through this. Basically, we have 3 types of hosts, again, from previous studies in this system. We have hosts that like to grow in the winter, so under the snow here. We have hosts that like to grow just upon snow melt, and then we have hosts that like to grow there, when like there's a lot of nutrients, a lot of light, and the temperature is way more, agreeable, let's say. And our question was like, if we look at viruses predicted to infect each one of these host groups, when are they active? So looking at the metatranscriptomes, not just like when are they present, but when are they expressing their genes to the point that we think they are producing new viral particles. If you look at this dynamic for the one infecting host in the spring, each line here is a single virus, and you can see like, it's kind of changes through time. Basically, the peak of activity for these viruses tend to be June and September, when we expect the host to be active. So the host is kind of growing and active, and the virus is growing and actively expressing their genes. Fine. This was the weird part. If you look at the one predicting to infect the host in the snow melts, they are not active during the May period when the host is supposed to be active. The peak of activity is delayed. It's definitely active more in June, some of them even in September. And it continues. When infecting hosts under the snow, they are not active under the snow. We don't see expression of the phage genes under the snow. We see expression of the genes later on during the season. What this suggests to us is there is some kind of difference between phages infecting what we assume, or at least what we I mean, what we pretty much know at this point are fast growers, so bacteria that like to grow pretty fast here in like very nice conditions, versus this bacteria here, which presumably, and I mean, it's more than presumably, like in all likelihood, these are bacteria that grow pretty slow. You know, you don't grow fast when you're under like 3 meters of snow and not seeing light. That's not the thing. And so there may be a kind of a trade-off here for phages infecting these different types of bacteria, in the sense of like, if you infect slow grower, you may not want to be super active immediately. You may want to let your community kind of grow and, you know, to a carrying capacity to the point that then you can start to kill them and infect them. So again, I'm saying like you may want to, it's probably more like an evolutionary trade-off, but that's kind of how we interpret this. Basically, immediate activity upon growth may be counter-selective here because it may be a kind of a dead end for both the virus and the host. That's a hypothesis. We don't know for sure. My whole point here is really, that's the kind of data you can look at when you have multi-omics, metagenome, metatranscriptomes, again, with nice metadata and with, you know, predicting pages from metagenomes and then predicting the host in silico. That's the kind of inference, or at least hypothesis you can start to grow purely from sequence data, which to me is very exciting. And if you want to know more about this project, this is a paper that just got published. So now I kind of built it up and told you like everything was amazing. Now I will kind of brush it all down because you will see how everything can be awful. So Yuna Wang, again, PhD student in the lab of Peter Girgis, had kind of similar data, slightly different. She's studying virus host interactions in deep sea microbiome mats. Absolutely beautiful. They have a robot that goes at the bottom of the sea, super colorful microbial biofilms, and then they take these samples, bring them back to the lab, and do metagenomics. And in her case, what she wanted was, you know, she looked at metagenomes to find the viruses and the microbes. And then she had 2 ways to link the viruses to the host. And we didn't have IPOP at the time, so it's kind of like backward, but it doesn't matter too much here. What she used is what's called CRISPR linking. I think everyone here is familiar with CRISPR at this point, but remember, you know, beyond the biotech tool, CRISPR is really an immune system for bacteria, and the bacteria will retain a small piece of the virus that infected the bacteria in the past. So we can use this as like a record of past infection, predict which viruses can get into which host. And then she also had Hi-C actually, so, you know, cross-linking of DNA, which should tell you which, you know, piece of DNA is with which other piece of DNA in the same cell. And that's kind of what the Hi-C is all about. And again, if you have like a piece of DNA from a virus and a piece of DNA from a microbe, and they are cross-linked together, one could assume, hey, I found a virus and a host, right? So it seemed very robust, like super strong CRISPR. Hi-C, I won't go through the technical details, but like Yuna did all the analysis right, and she did all this right cutoff and everything. She's really conservative in like these cutoffs. So everything is right. And then when she had all this data, she built the virus host network, right? So this kind of network here, the big symbols will be hosts. You have like the different types of hosts with different shapes. The small dots will be viruses. And you expect mostly something like this, right? One virus, one bacteria. I'm happy with this. And that's what we actually saw in the real data. And well, first it's super connected, which is like, oh, that's super cool. And then you start to see something you don't expect. This kind of thing especially. This is one virus connected to bacteria archaea. So either we found a virus that is able to infect both bacteria and archaea, which is amazing and absolutely unheard of, or we need to go back to the drawing board and think a bit more. And in this case, we don't have technically the answer, but I don't believe we have any virus that can infect bacteria and archaea. What we have to remember is anything we use, especially CRISPR and Hi-C, is about which piece of DNA is in which cell. It's not the same as infection. We use that as a proxy for infection. It's not the same thing. You could have a virus genome in a cell without ever having a successful infection. It can be here by mistake. It can be here because the cell exchanged cytoplasm with another cell that is nearby. And that, I think, that's my personal hypothesis, or at least my preferred hypothesis. Yuna kind of drew this really nice figure at the end of the paper that kind of details what could happen here beyond infection of super broad host range. One of these is because these are biofilms, we have symbiont partners, and they tend to exchange cytoplasm. And what we see a lot in this kind of cross-infection, cross-predicted infection, I guess, are usually between partners that we expect have very, very tight relationships, like quasi-symbiotic. And so we suspect that there is some exchange of cytoplasm that happens sometimes, and that, you know, is enough of phage genomes to go from one host cell to a non-host cell and still be like, you know, lead to CRISPR acquisition or like linked to Hi-C linkage. Or you could also just have wrong infection, like, you know, out of the billions of virus particles, one of them is bound to just inject its DNA into the wrong cell. That will happen. If this happens, you know, in a frequency high enough, again, strongly structured biofilms. So you will always have the same bug next to the same different bug that tend to co-occur. I think that's kind of not completely out of the realm of possibilities, that it's happened frequently enough for us to capture this signal, basically. But my bottom line here is, this is all prediction from bioinformatics. When you look at this, and especially when you find something absolutely amazing that no one has ever witnessed before, we should always kind of think about like potential methodological issues or biases or answers or explanations beyond or before kind of jumping to like, I found the best virus ever. And again, CRISPR hits are not always between phages and hosts, as far as we can see from here. And if you want to know more about this, just again, this is Yuna's paper that kind of goes in much more details about what we found there. Okay, so quick conclusion of the viromics. To me, I have this as a title here, it really opens an incredible perspective at this point. I'm old enough to remember the beginning of metagenomics with what we call next generation sequencing at the time. So, you know, I'm the generation of like starting with 454, right? So I remember the small pieces of genes and everything, that was cool. But seeing the rise of genome assembled from metagenomes has been amazing, especially for viromics, I would argue. Because again, these various genomes, which we had really no access to before, now they just fall out of the assembly, you know, pretty much readily. So it's all about like what we can do with them, how we can enable, you know, create more tools and databases to kind of understand better how they impact, how these viruses impact microbiomes. Again, purely starting from the sequences. Host prediction, again, I show it as like one of the key missing pieces. I think we are getting there. We have still a long way to go to be able to robustly predict hosts for each and single, every single virus we see in this data. Part of it is a technical limitation of the bioinformatic tools. Part of it will be biology. Biology is messy. So what we think of like, it's clear like host range, like for some cases, we won't know for sure if the virus is infecting this cell or this cell successfully. If it's found in both, it's just found in both. And again, I do think at this point, we have this framework that is sufficient to get into this complex and large-scale virus host dynamics, especially if you have time series, especially if you can have multi-omics, especially if you can link this to really nice metadata for which you have like a lot of information around each sample. That's really where the power of viromics will kind of shine, essentially. So that's kind of what we do at JGI, but also what we kind of try to promote as like, these technologies are mature and ready at this point, I think, to put into everyone's hands. And if you're doing any kind of microbiome work, it's something I think should be considered if you want to kind of have any kind of overview of your system and how it changes and what happens in there. Now, of course, and I will have this, you know, use the last few minutes to pitch a completely different projects. But of course, for all this to happen, what you need is a lot of, again, multi-omics. I pitched you like, you don't want just metagenomics anymore. You want metagenomics plus everything else. You want metatranscriptomics, you want metabolomics, you want all of this on the same samples with very nice metadata. If anyone has tried to do this or tried to do this kind of analysis from samples you did not collect yourself, this is usually you and me. And that's why, you know, something that was recognized by some people at JGI a few years ago, this idea that if we want to really leverage fully this metagenomics and multi-omics framework, especially the reuse of public data, we will need much better standards in terms of how we share data and how we share metadata. And that's kind of one of the bases from which this National Microbiome Data Collaborative was established. This is a DOE-funded at this point collaborative that really means to bridge this gap and kind of, you have the vision statement here, so connect data, people, and ideas. So essentially, making it way more easy for everyone to understand, okay, which data is where, and what other data can I find on the same sample, and what are all the information I know about this sample. Seems simple, but if you ever try this, it's really impossible. We have a lot of things going on in MDC. We have different tools and everything. I won't go into this, but the last slide I have, and the last thing I want to pitch here is, as part of NMDC, we have what's called an NMDC Ambassador Program. This is really some program dedicated to PhD students and postdocs who have, you know, either they are passionate about metadata or they have been frustrated by metadata or they want to learn more. And the idea is 1 year program, you get a stipend for like a few hours of work each month. The work will be training in how to best collect and use standards for your metadata and training on how to actually run a workshop or training yourself to teach others about metadata. And at the end of the year, you also kind of host your own workshop. You kind of get all this also cohort learning and all that. And so we have the next cohort, which will be announced in January with application due in February. So since we have a lot of students here, I wanted to share this. To me, it's an amazing opportunity. I have had the privilege to work with this last cohort, and this was very nice. And again, same as for JGI, if you have any question about an MDC, please come and find me later today. And with that, thank you.
I love the I love the pitch for multiomics. In terms of sequencing technology, what gives you the best viral assembly? Is it the new high fidelity long reads, or is it depth of coverage from Illumina?
It's both, but high fidelity long reads has been really great in our hands as well, and in the hands of many. To us, the main issues has been that it still requires really high quality DNA extractions, which may or may not be possible in your samples. If it's possible in your sample, I would argue, yes, definitely think of this and consider it at least making one long read sequencing for your system to get really nice reference genomes. And then if you want to do like, time series or whatever, like do the Illumina, short reads time series, and then you can combine these two technologies to get both like super nice reference genomes and high resolution temporal data. That's kind of the model where we are trying to go to. One switch to that is if you can't have good DNA extraction and you can't get long reads, what we do nowadays is a lot of combined assembly of the short reads. So kind of faking the deep sequencing, right? Let's say you have like a 50 samples time series. We can take the 50 metagenomes, each sequence at like, 100 million reads. We can just smush them together and do one big assembly. That's a program called MetaHipmer. Initially, I was like maybe worried about like, will this make chimeras and whatnot? But we carefully benchmarked it, and it looks like it's doing way more good than bad, basically. And in our hands, it has been extremely good, especially when you have very complex systems like soil, where you have very fragmented assemblies still from the microbes and the viruses. Just for Clément's paper, that's what we use to get very nice phage genomes from these 48 metagenomes all co-assembled. So I would say that's kind of your two approaches to get better assemblies.
Oh, just a quick one about long read sequencing as well. I'm just wondering if you've been able to look in your data sets for any modifications on phage DNA.
Yes, and in a few cases where you can. So it's even worse for phage DNA modification because you need not only to have very good DNA extraction, you need to be such a high amount that you don't need to do PCR. And lots of our libraries still need some PCR to be kind of, taken to the level that they can be sequenced. For the few cases where we have enough DNA, we have looked at this. For some residing prophages, you can see that you have the same methylation mark between the host and the phage. For many viruses, though, it doesn't seem to be the case. And it seems like restriction modification are way more used as a defense mechanism to the point that basically you don't have a good match between the phage and the host modification. They all use different marks basically to kind of counter each other.
Great, thanks.
Thank you very much for the talk.
Thank you.
Our next speaker is Jose de la Torre. He's an assistant professor in the Department of Biology at SF State. Full professor. My bad. He received his PhD in biochemistry and biophysics from UCSF. His lab focuses on the ecology and evolution of microorganisms living in high temperature environments, particularly hot springs. The lab is particularly interested in how environmental conditions, chemistry and pH, and evolutionary history and the influence of composition and function of microbial community in these extreme environments. Welcome.
Thank you. Thank you so much. And thank you to the organizers for inviting me. If I start to shake, it's because I may have some PTSD. The last time I was in this room 26 years ago, I was defending my thesis. So, anyways, moving on.
anyways, so thank you. I'm going to tell you a little bit about this project that follows perfectly with what Simon just talked about. Although I am old enough to remember when metagenomics was Sanger sequencing of phosmids and backs. So I really feel a little bit old compared. All right, so this project is really sort of a side project that started as a pandemic pivot, which most of us have had in our labs. But it's turned out to be really interesting. And so it's still a little bit preliminary, but hopefully I'll tell you something about the biology of this system. So our lab, let's see if I'm going to get the buttons right. Our lab studies archaea primarily. For those that don't remember, archaea are one of the 3 domains of life, although that is a changing proposition as we speak. And in particular, we study a group of archaea that were first detected using 16S ribosomal RNA gene sequencing by Jed Furman's lab at USC and Ed DeLong then at in Woods Hole. And these organisms were found in seawater. And this was revolutionary because until that point, most of the archaea had been found in extreme environments. As it turns out, further studies, again, cultivation independent studies, found that these organisms were extremely abundant in marine systems. They could account for up to 40% of the bacterial plankton below the phototrophic maximum. This is data from Hawaii showing that below about 100 meters, you really have a ton of these organisms. And so some people have made the argument that these are some of the more abundant organisms on the planet. At the time, this is 2001, no one had any idea what these organisms were doing. And it wasn't until the advent of metagenomics that we started to get a clue of what these organisms do. And it turns out that they play an important role in ammonia oxidation or nitrification in the marine environment. This is a key limiting step in the nitrogen cycle. And because of their sheer number, and it turned out that they're also autotrophs, it was then hypothesized that these organisms play a big part in driving at least the marine carbon and nitrogen cycle. Fast forward about 10 years or so, and we now know that these organisms are present in a wide variety of environments. They're found in marine environments and freshwater, host associated primarily with invertebrates in marine systems, but we've also apparently, groups have found them associated with human skin. They're very abundant in soils as well, some extent in wastewater treatment plants. And then in 2008, we found them in hydrothermal systems. And so what I'm going to tell you about today is mostly our work in these terrestrial hot springs. This is a picture of the hot spring where we first identified the very first strain of these thermophilic ammonia oxidizing archaea. The organism is pictured up there. It's fairly nondescript. But what we found was that these organisms were widely distributed worldwide in hot springs that are near neutral pH, slightly alkaline, at temperatures above 65 degrees. We can detect them all the way up to about 95 degrees, but they really prefer a temperature around 70 to 80. They seem to form a deep branching group within all of the ammonia oxidizing archaea, separate from the mesophiles. And there seem to be a couple major lineages that are primarily biogeographically distributed. And part of the reason, and we now have cultures from a large number of these hot springs around the world. So we have some from Yellowstone, we have some from northern Nevada in the high desert, and we have some from China as well as part of a large multi-center project funded by the National Science Foundation. And if we look across the world, we have lots of strains now. We also have lots of metagenomes, lots of metagenome assembled genomes as well. that we can use. And our initial idea was to look at these, because we were primarily interested in physiology, was to compare these organisms to try to get a sense of what the shared genes were, and then establish the basis for their electron transport and energy metabolism. But what I'm going to tell you today is more of a study of island biogeography. And so many of us use hot springs, terrestrial hot springs, as a terrestrial analog for islands. Island biogeography and ecology is a well-established principle where you can look at organisms that are essentially separated from each other in space and time with barriers to gene flow. The idea is the same in hot springs, that you can use hot springs that are physically disconnected to look at the evolution of organisms and natural communities in those systems. Whether that's a real valid assumption remains to be clear, because we don't know what the underground plumbing is, right? So there may be connections between these. The worst case scenario is that actually gene flow is mediated by research scientists who don't clean their sampling equipment between springs. This is a real problem, actually. You know, it's funny, but sometimes not. All right. So when we started comparing the genomes of these organisms, what we found was, so this is a standard pan-genome plot. And so this one was done by Brittany Baker, but a similar analysis was done by Carlos Gomez here in the audience. What we found was that the core genome of these thermophilic ammonia oxidizing archaea was very stable. They had about 1,000 genes or so that were stably found in all of these organisms, regardless of where they came from. This is 1,000 out of only about 1,500 genes. So about 2/3 of the genome is reliably found in these organisms. And when we started looking at how close these organisms were to each other, what we found was within a geographic region, the core genome had a really high level of nucleotide identity. Okay, so within our Chinese strains, it was about 99 to 100% identical. The European genomes are also about 99 to 100%. The Yellowstone ones are the same. The ones from Nevada are the same. Across those geographic boundaries, there's a significant breakdown. So these different classes of organisms probably reflect different species or genuses in different places. Whether that's true biogeography or whether it's simply small, imperceptible changes to the environment that we're not good enough to detect that determine what grows and what doesn't, the fact of the matter is that when we bring them back into the lab, we find different species using identical culture conditions. The genomes themselves are also remarkably conserved in their synteny, in the gene order. And when we look, so these are all our Chinese strains here. And what you can see is that the gene order, so these dark bars, those are the genomes. These light gray bars are regions of similarity. And what we find is that for the most part, the gene order is identical. There are clear areas of differences. Some of those are integrated mobile genetic elements like these guys. And then in some places, we have some regions where we have integrated genomic islands that are always integrating in the same part of the genome. This has been seen in bacteria before, in other archaea as well, and are referred to as replacement islands or flexible islands, where you find gene cassettes that swap in and out. In our North American strains, what we see is that those are, again, we see some mobile genetic elements, but we also see glycosylation islands that are different. This is a typical pattern that we see, and presumably is a defense against viral predation. By changing glycosylation patterns of surface proteins, organisms can escape viral predation. And so we see a lot of those types of changes. We also see some metabolic changes, like the presence of a urease that can cleave urea into ammonia that can then be used for energy production. And again, these are patterns that have been seen in a number of other papers, whether they are these integrated conjugative elements that hop in and out, or other types of flexible islets. Wow, this is really sensitive today. All right. Those conjugative elements, we find them all over the genomes of these organisms. On average, each organism has about 2 of these presumed ICEs. They're different than the typical bacterial ICEs that have been most described because we can't find any type 4 secretion systems involved in the formation of a physical pilus that would exchange these elements. But what we find is that these elements are extraordinarily conserved across geographic boundaries. So this particular element that we see here, Brittany found it in every single genome in China and in Europe, actually, and even in Japan, in a gold mine deep underground. So these elements have been essentially immobilized within the genome. One hypothesis is that they're essentially been locked into the genome through the loss of an integrase or something that allows them to work like E. coli HFR strains. So for those who don't remember, HFR strains are strains that have the F plasmid integrated permanently into the genome so that it then, instead of transferring just the plasmid through the pilus, it transfers portions of the chromosome as well. And that's a mechanism for maintaining homogeneity of the genome. So that may be one explanation. In one of our strains, our type strain, we actually found a plasmid that mobilizes. So we can see it, we can detect it both in the genome and in the cytoplasm and also in the supernatant of the culture as a DNA circle. And this element is strange. We can see it both through read depth, tetranucleotide signatures, and codon adaptation indexes. It looks different than the rest of the genome. At this point, we're still trying to figure out what this element does and how to harness this to do gene expression, heterologous gene expression. It's difficult to work with a 50 kb plasmid, but our hope is that we can use this. And we're not sure how this plasmid is transferred, but this is again reminding you of how conjugation works. But what we think is actually, because this has no pili, we think it's actually mobilized through vesicles. And vesicles are one of the hot topics in microbiology, as many of you will know right now. We can find vesicles that have different payloads, proteins, RNA, DNA. And what we found was that in a DNA protected compartment in the supernatant of our cultures, we can detect the circular form of this plasmid. And so our initial hypothesis was that the vesicles mobilized that plasmid, they fused with new cells, and then the plasmid integrated into the genome and then repeated the cycle by forming new vesicles. Unfortunately, that doesn't seem to be the case. Instead, this seems to be a stochastic mechanism. What we found when we sequenced that vesicle fraction is we found essentially the entire genome present in the vesicle fraction. The plasmid is definitely there. Apparently, somewhere along the line in our cultivation efforts, we lost a provirus, which happens. Nature selects. But the entire genome was present in the vesicle fraction, and the plasmid was not particularly enriched in the cellular fraction compared to the extracellular vesicles. And so instead, what we think may be happening is that these cells may be actually secreting vesicles containing plasmid and DNA. Those vesicles may fuse, and this is another mechanism for horizontal gene transfer, both of the plasmid, but also of chromosomal elements. So this would be a fourth mechanism in addition to transduction, transformation, and conjugation. But at this point, we have not shown that these vesicles actually fuse and recombine. We're trying to do that now. What is interesting is that when we look at metagenomes, we can see that some of these elements are very geographically specific. That element that I was just showing you, we've seen it in metagenomes only in Yellowstone. More recently, we're starting to see it using the software that Simon was talking about earlier, Genomad and IP Hub. We found similar elements in other metagenomes in Europe, which suggests that the software is not only just detecting viruses, but also mobile genetic elements. The boundary between those has become really fuzzy. But what we found was that these metagenomes could be a good source of assessing microdiversity. And so what we have here is what we call a read recruitment plot, where we've taken metagenomic reads. Each of these points represents a single read that's been that matches a reference genome. So down here we have a reference genome, and it's been plotted on this plot by its position, the position of its match, and the percent identity on the y-axis. And what we can get is a nice picture of genomic islands and also the types of organisms present in the environment. So in this particular case, this reference genome doesn't have a close match in that environment. There are very few reads at 100%. Most of the reads are down here at 95, 96%, suggesting species level divergence. And we can clearly see regions in the genome that are not represented in this population, suggesting that those are genomic islands. I'll skip ahead a little bit for a second. And so we can see different types of patterns. So here's, for example, a pattern where our reference genome is a very good match to the metagenome. Most of the reads that map are at 100% identity. It's just hard to detect those when they all fall on a single line. And others have broader distributions where there's more variety in those. And so in the pandemic, what happened is this became fodder for a student who decided that because she couldn't get back into the lab, San Francisco State is particularly risk averse. And so this master's student couldn't get back into the lab for 9 months. And so she came up with a project to look at microdiversity of the populations based on recruited reads. And the questions she was asking, and the questions that we're still asking, is which genes and pathways in these organisms show the highest level of microdiversity within a natural population. So as opposed to comparing isolated genomes, which is one organism from a population, can we look at the metagenome as the total sum of that population? Do those patterns of genetic variability match when we look at different hot springs or different temperatures or different environments? And can we understand something about the selective pressures of the organisms that are in these environments? And so this project was started by Rebecca, who's now in Ann Dekas's lab doing her PhD. I should say that all of this work is done by undergraduates and master's students. And we have a large number of data sets from North America, many of them from the Joint Genome Institute, but others that we generated as well. And the idea basically is that you recruit reads to a reference genome. Choosing that reference genome carefully is very important. And then those recruited reads, then you can look at how many differences they have compared to the reference genome, calculate the SNP counts, and from that generate what the nucleotide diversity for each gene along the genome is. And when we do this, what we see is a large variety across the environments we've looked at. Some Some springs, such as this spring in northern Spain and a few springs in Yellowstone, show very little diversity of that population. And this is using reference genomes assembled from those metadata, from those metagenomic data. All right, so they're relevant genomes for that population. Others have very high levels of genetic diversity within that population. And we start getting into concepts that I was never trained in here at UCSF in terms of ecology and evolution, things like effective population sizes, things like that. We don't have a good understanding of what's happening within these populations. And so we've looked within these to try to tease apart data. So here's an example where Rebecca looked at different categories to see if certain types of genes were more diverse than others. And for the most part, there wasn't a strong pattern. We focused on a couple springs that sort of illustrate the main patterns. And we've looked at the core versus the variable genome. And again, we don't see a strong pattern. Always there are outlier genes, but we don't have a good sense of, there isn't a clear pattern across these different categories. And so what we've started doing is focusing on these outliers. Here's another example. Oh, this came out blurrier than I hoped. But where we have looked at the number of non-synonymous versus synonymous mutations to see what the pattern of selection is. And as a general rule, this black line here indicates a ratio of non-synonymous to synonymous of 1. What we have in general is evidence of negative selection, so purifying selection, where the genes are essentially being held back from changing their amino acid sequences. And that seems to be true across most of the genome. We're trying to tease out the few genes that are clearly outside that distribution pattern, but most of those are hypothetical, which is the bane of our existence always. But we're trying to do this a little bit more consistently and look across springs to see if those genes are actually telling us about the selective pressures, and especially what type of environmental factors are affecting these patterns. Unfortunately, as Simon alluded to, we do not have good metadata for most of the metagenomes that were uploaded to IMG or to NCBI, which makes analysis difficult. And so in the last second, what I'll talk about is a plan that we're trying to put together. We're hoping to get JGI on board on this, but this is a site in Nevada that's already been well studied by the Joint Genome Institute. There's 3 really deep metagenomes for this site here at 85 degrees, at 70 degrees, and 60 degrees. We've been working on this spring for a number of years in collaboration with Brian Hedlund's lab at UNLV. And what we'd like to do is start a time series. And so one of the students from the RAMP program that is spearheading is involved in this project. And what we're trying to do is get good quality, high molecular weight DNA in enough quantity to do both all the things that Simon was talking about, sampling once a month with good geochemical data as well. And so that's where we're trying to go. Claire is the RAMP scholar working on this, and Jill is a master's student that both just started this August. All right, and so I'll end there. And these are a number of students that have worked on the project. It was started by Rebecca, and most of the data that I showed today was Rebecca's data and Andreas Williams' data. Andreas is a phenomenal bioinformatician looking for a job. He graduated, and so if you have a job for a great bioinformatician, Andreas is your man. All right, thank you.
okay, you kind of set me up for this with your island biogeography, but have you just looked at the variation among your springs in terms of diversity, standing diversity? Does it have to do with how many springs are nearby, like what the dispersal pool would be?
So if I'm understanding the question, really, it's how does the diversity vary with distance from each other? And we've tried to do that. The best data that we have is actually from our samples in China, and the ones that are closer together are definitely more similar together, but the amount of microdiversity is variable within those. And so I think it has more to do with the size of the pool itself, which may have something to do with the size of that population, and possibly how many different micro niches that we aren't detecting actually exist within that system. We're obviously much too simplistic.
I guess a similar question to Brett. Are there sort of known animal vectors or, I don't know, geographical processes that transmit these archaea between different hot springs in the same location?
In Yellowstone, we always worry about bison trudging through the pools. It's usually bison, not bears or things. There aren't known vectors, but like the Colorado watershed that Simon was talking about, a lot of these systems are flooded with snowmelt in late spring, and some of that may spread organisms across, although it's clearly at a much lower temperature than they would prefer. But again, the main concern I have is underwater plumbing. In the Nevada system, anytime you drill a hole, you create a geyser, literally. And so there's clearly a lot of water underground, only about a meter down. And so we have, and it's under pressure, so it's circulating. We don't know. But this is why we need to talk to geologists more.
All right. Thank you for the excellent talk. You are an associate professor. Yes. All right, and with that, we have the last speaker for this session is Dr. Britt Koskela. She's an associate professor in the Department of Integrative Biology at UC Berkeley. She received her PhD from Indiana University and is currently focused on understanding how bacteriophages shape bacterial evolution, microbiome diversity, and ultimately the health of host organisms. The lab is currently also interested in how plant microbiomes can shape relationships between host and pathogen. And with that, please.
All right. Yep. Look at that. All right. All right. Great. Oh, I'm going to start my timer as well. So that All right. Wonderful. Thanks for the organizers for the invitation. It's always nice to come across the bay, especially when the bridge is working. And also to those online, thanks for joining. Okay, so I am indeed interested in phage and have really been enjoying the talks that are relating to viruses, but I will not be speaking about phage at all today, sadly. So if you came for the phages, come talk to me at the coffee break. I can tell you what we're doing in that space. Instead, today, I'm going to focus on some of the work we've been doing on the plant microbiome in disease and health. So when you think about the plant microbiome, you might think about the rhizosphere below ground, and you would be correct that that is where most of the research has happened and happens. But there's a huge wealth of biomass, 60% of land biomass, which is the phylosphere, the leaves and stems and flowers of plants above ground. And this is remarkably understudied compared to the rhizosphere, but also hosts a really important microbiome, as I will show you. But actually, the main reason I started working on it is because I'm an ecologist and I wanted a model system. I'm unapologetically a fan of plants as model systems, less funding, but they stay put and there's less ethical concerns. And a lot of the questions that we ask, it's just a host, like any other host, whether it be a human or a mouse or a plant. And so the plant phylosphere has a couple of amazing advantages for me thinking about the microbiome through an ecological lens. One of them is that it's beautifully spatially explicit, right? I know where a leaf starts and ends physically. And so I can, going back to this island biogeography idea, right, it's very easy to say something is sympatric or interacting locally versus allopatric from a different population or a different community. And a lot of what we do rests on that idea. It's easy to measure and manipulate. So if you've ever read about fecal microbiome transplants, I'm going to show you how we do these on plants. We do leaf microbiome transplants, and it works amazingly well. You can recapitulate natural microbiome diversity simply by spraying microbes onto leaves. There's clear trait variation within and across species, right? Every leaf is a little bit different. This is my favorite part of it, because it is essentially a sort of petri dish-esque organ. It's highly culturable. The vast majority of the microbiome is culturable. We don't need anaerobic chambers, et cetera. So it's a good model. I'm happy to talk more if anyone's thinking about switching over into the plant world. But also, and what I'll talk about at the end of today's talk is that these microbiomes do indeed have an important function, which again has been pretty understudied. This is a fantastic review. If you need further convincing at the end of my talk that the phylosphere is important, this takes a beautiful overview of all of the different roles that the phylosphere microbiome plays in everything from plant function to biogeochemical cycling. But in the lab in particular, we ask questions on the microbiome side that are very similar to questions you might ask in any microbiome. So how do microbiota get there? How does community assembly happen? How do microbes within the microbiome, and in fact, the whole microbial community, adapt to their host over time? Can you see evidence of evolution within a host and ecological selection as well? And then how does the phylosphere impact health and disease? And ultimately, we want to leverage this knowledge to increase host health and agricultural sustainability. Okay, so when we think about how a microbiome gets there, we often think about how it got there in a natural setting. So we work on tomato, as I'll introduce now, as our main model organism, although we have quite a few other, we work on pear trees as well as our natural system. But most of our work is on tomato microbiomes. And so when we think about how a microbiome gets there, how a community assembles, I think about a tomato growing in nature. So this is a little Solanum pimpinellifolium growing in Peru. And you could think about all of the ways that a microbe could arrive on a leaf. And you might imagine, and I'll show you that you are correct, that this can happen via seeds. So none of these plants emerge as seedlings sterile, if you will, just like humans. There is also then horizontal transfer from wind, insects, soil, and a lot of that will be transmission from neighbors, so proximity to others. All right, so those were at least the three easy to come up with mechanisms of how a microbiome might assemble. Let's go through and find out whether these are happening. So the first is just I'm going to show you a small snippet just to convince you that tomato seeds are indeed hosting their own microbiome. This is work done by Norma Morella, who was a PhD student and is now at Pivot Bio, and Shirley Zhang, who's now a PhD student at Cornell. And this was Shirley's undergraduate thesis. And I'm just showing you here the sort of premise for her study, which is that if she goes out to UC Davis, an organic farm, and samples tomatoes, surface sterilizes the fruit, and then looks at the seed, those seeds, both epiphytically and endophytically inside the seed, have microbial communities. Now, when you do 16S sequencing, what you find out is even though they're somewhat diverse in the sense that there are multiple ASVs or taxa there, it's almost all these Pentoa species. And this has been, we've repeated this numerous times, this does seem to be the dominant seed-associated bacterium. Now, I won't show you this data here, but critically, this pretty low diversity and definitely low density microbiome of seeds does indeed work to colonize seedlings as they sprout. So that becomes the dominant bacteria in early seedling development. And we've shown in the same paper that these microbes are disease protective. So just that small diverse microbiome that's there right away when a seedling establishes is enough to prevent Pseudomonas syringae, one of the major pathogens of tomato, from causing disease. So certainly this is important both to seedling, but, and this is again relevant to any microbiome you work on, that early establishment also probably plays an oversized role in future development and assembly of the microbiome. And one way you can think about this is as a priority effect. And priority effects are very well known in ecology. This is a beautiful review paper by Rena Debray, who was a graduate student in the lab and is now a postdoc in the Max Planck Institute. And what this paper did was think about all of the different ways arrival order of bacteria might change future community assembly. So you can have facilitation, where once something's there, something else is more likely to establish, or preemption, where once something's there, something later arriving is less likely to establish, and so forth. And if you're interested in this, it's a great review. And I'm going to show you one of the neat things that Rena did is reanalyzed a data set from the infant gut, the human infant gut microbiome, and characterized these beautiful time series data sets into little snippets of persistence for particular ASVs. And this method's developed in the paper. If you're interested, please reach out. But what she did is this is just one example. She would go in and look at specific taxa, and for each taxa generate a metric of persistence, a measure of persistence. And so I'm showing you the three examples of persistence here. The top one is where it arrives and was present at each time series during that little window, and it's a sliding window. The next one, right, we have a persistence that's a bit lower. It was there for a few times and then petered out. And then finally, you have something arrive but not establish. So she got these measures of persistence and then asked whether you can predict persistence based on the microbiome at the time point before it arrived. And the answer was resoundingly yes. And so what you're looking at is persistence, is the colors here. And hopefully you can see that they sort of separate out. This is just a classic PCOA plot of microbiome diversity, which I think in this audience, I don't need to spend much time explaining, but basically suffice it to say that different microbiomes were more likely to allow persistence of this particular taxa than others. All right, so that's just one reason of many that we really should think and care about that initial assembly, the initial microbes that are there in early development. But if you're thinking about plants, I've already said that what we think about is what happens here, but in reality, what we're dealing with is what happens there. In a greenhouse setting, in monoculture, in age-structured huge fields, and that's likely to look a lot different. I've already shown you that seeds are a transmission mechanism. Well, the majority of seeds in agricultural settings are sterilized to prevent seed pathogens. So that's already disrupted that first major mechanism of dispersal. And Eli Melferber, who was a PhD student and is now a postdoc at Georgia Tech with Sam Brown, Eli built a beautiful synthetic community, which I'm going to come back to at the end of the talk, of the tomato phylosphere. But one of the things that we discovered early when he was running his experiments is that we also, in all of our experiments, we have control plants where we don't inoculate the phylosphere above ground with any microbes. And we randomly scatter them, of course, along with our experimental treatments. And what I'm showing you here is only our control plants. And if you do 16S sequencing of our control plants, you might think they have a normal and diverse microbiome. But one of the things we do in the lab is use droplet digital PCR to estimate absolute cell numbers of bacteria. And when we use droplet digital PCR, what we see is that even though, yes, there's some diversity there, the actual cell numbers in the phylosphere of greenhouse grown tomato is very, very low. And I'm going to contrast this in a moment. So take my word for it that that's low for the moment. Okay. So it does seem like the greenhouse is a place where, if you have shelter plants from wind, insects, rain, that you've also disrupted the second mechanism of transmission. All right. So far I've rapidly gone through, you can get it through seeds, you can get it through wind, insects, and rain. What about from neighbors? And I don't know whether anyone has read that old roller derby story about roller derby players having microbiomes that are skin microbiomes that are more similar after a match than before. Well, this is like the plant equivalent of the roller derby story, all right? So this is work led by Kyle Meyer, who was a postdoc in the lab. And what Kyle did was, in an experimental plot right near our lab at UC Berkeley, we took up a city block and created these little mini plant neighborhoods outside. And in each neighborhood, we changed who the focal plant was, whether they were tomato, pepper, or bean, and who the plant neighbors were, tomato, pepper, or bean. And we did this in a huge randomized block design. And at the end of it, we asked a couple of things. Oh, I should say, we also did this at 3 different time points. And at each time point, harvest 1, we harvested the focal plant only, and then replaced it with the same species that we just took out at the same age it was when the experiment started. So we kept the focal plant more or less the same age through time, but we allowed the neighborhood to grow up around it. Right? The data were remarkably hard to analyze and think about what this would actually look like. But what we came up with as the cleanest way to think about this problem is when you take all the variation in the microbiome and you try to explain variation by host identity, whether the focal plant is tomato, pepper, or bean, that gives you this adjusted R-squared. This is the amount of variation explained by host or by the neighborhood in which it is in, or by the block, the noise, right? And what we found is that when, at the beginning of the experiment, when the neighborhood was quite small, the focal plant itself was very good at filtering out the bacteria that landed in the phylosphere. And so the primary factor explaining microbiome diversity in the beginning of the year was whether that focal plant was a tomato, pepper, or bean. But as you allowed the neighborhood to grow up, you actually at the end had a higher neighborhood effect than a host identity effect. And this is another way of saying, in a more general sense, that dispersal matters to microbiome assembly, and that although a host organism is able to filter and affect who survives from those dispersing species, at some point that mass effect of dispersing organisms might overwhelm the ability of an individual to filter. And so, for example, if you were swimming or drinking muddy water constantly, that might reduce the ability of your gut microbiome to actually selectively filter who's there. All right. So communities assemble through all of these processes, and I'm going to argue that all of these processes are disrupted in agriculture. So what do we do with that information? Is there any way that we can get ahead of this? And at the end, I'm going to introduce the synthetic community. But before that, I want to introduce an idea that I hope will be relevant to microbiome researchers across systems. Because one of the most fun things we've been doing over the last couple of years is thinking about how you can select or experimentally evolve a whole microbiome. And this work was started with Norma Morella, who I've already introduced, who's now at Pivot Bio. And what Norma did is she went out to the field and collected a diverse tomato microbiome, just one that was generated from many, many plants, pellet, wash, and sequenced the heck out of that initial inoculum. And then she sprayed replicate plants of 5 different near isogenic lines of tomato with this inoculum. And importantly, we sprayed them multiple times for the first 8 weeks at about the equivalent bacteria that you find in rainwater. So really quite low density microbial inoculum. And we have slowly allowed the microbiome to assemble over those repeated inoculations. And then at the end of that trial, that 8 weeks, we would take just the phylosphere above ground from a single plant, shake off all of the microbiome of that plant, pellet it, and that would become the inoculum for the next generation. And so each of these lines is independently evolving. There was no mixing across them. We did that for 4 transfers or experimental passages. And then the question becomes, well, and then we use droplet digital PCR to measure absolute abundance and 16S sequencing to figure out how composition was changing over time. And we ran the experiment and we're quite excited because we had stuff at the end, so we did something. But our first challenge was figuring out if it had worked, if it was a fair test of what we were trying to do. And the way we convinced ourselves that it was a fair test is by looking at two things. So the first is we sequenced the original inoculum. We knew which taxa were there at the start of the experiment. And then we can look at each transfer of how many taxa were there from the original inoculum. And that's this dashed line here. And what we found is that through selection, we selected out about 70% of the taxa from that original inoculum. So they were gone. Now, the worry is that they could have just been replaced by other things flying around the greenhouse, right? So the bars there are showing you what proportion of the bacteria we found on the leaves that were from the original inoculum. And you can see that that's upwards of 90% even at the end. So we really did select out a lot of taxa, and the remaining taxa were the dominant members at the end of the experiment. So that was a huge relief and really exciting, but then got us to our next challenge, which is how the heck do we show whether a microbiome adapted? And that we kind of got stuck on that for a little while. And what we came up with is using community coalescence theory, or community crashing as we call it in the lab, to test what happens when you throw an unselected microbiome and a selected microbiome together, who wins when you put them in head-to-head competition. And we use live dead droplet digital PCR to create an inoculum that is 50% live cells from just the first passage, so greenhouse equilibrated but not adapted, to the end passage. We put that inoculum together and we spray plants with either the mix or either the inocula neat. All right, you with me? All right. At the end of that, we just use 16S sequencing to ask what happened. And what I'm showing you here are just the two pure lines. And the first thing to say is this is the first time plants were in the same greenhouse at the same time with these different inocula, because of course previously they were temporally separated. So it's very nice and relieving to see them once again segregate out so nicely. Indeed, our selected microbiome in purple looks very different than our unselected in orangey color. So then what happens to the 50-50 mix? It was statistically indistinguishable from the selected microbiome. So another way of saying that is our selected microbiome was uninvadable by all the things that we just selected out, which is pretty neat. And once we had this, this model established, then the next question is like, what are we going to do next with it? Because you could imagine doing so many different things. But in the end, I'm an ecologist. I'm actually a disease ecologist by training. And one of my long-term interests is generalism and specialism. Right? Why would a pathogen or a bacteria specialize on a host organism rather than having a broad host range? And so I've already introduced Kyle Meyer here, who led this incredible effort that I'm about to show now. We wanted to ask whether that generalism and specialism can be true at a whole microbiome level as well. So we took the same approach where we do these experimental passages, but we either experimentally passaged the microbiome, which by the way, all started from tomato initially, so a highly diverse tomato inoculum. We passaged it for 6 passages in this case, so a bit longer, either on tomato or one of the wild tomato cultivars, Campanellifolium, or bell pepper or bush bean. So these are on the top are what we call the conspecific transmission lines. So every generation or every passage, that microbiome stayed on tomato or stayed on bean. And then we had lines where we swapped hosts every generation, every passage. All right. And the question was, can we get specific adaptation to hosts up here? And when we don't allow for, a sort of continuous host environment, do we get something resembling generalism? Again, how to analyze such data was a real challenge, or how to test the hypothesis. And I've already introduced the idea of using this R-squared value of host identity to determine how much host filtering or how much host specificity exists. And so when we apply that same thing, here are our different passages. So we 16S sequence everything and ask how much variation in the microbiome can be explained by the host species identity. And when we do our vertical transmission or our conspecific lines, they filter out quite well. So you can always tell what's a tomato microbiome, a bean microbiome, a pepper microbiome. But in our host swapping lines, that decreased. So these became less host specific over time. So then the flip question is, well, did the vertical lines become highly adapted to their host? I've already introduced the idea of these community crashes, but to test that, we basically took our conspecific pepper line, for example, our conspecific tomato line, crashed them together, 50-50 live cells, sprayed them on tomato and asked who won. And hopefully this won't come as a surprise to you at this point in the talk, but basically, if you just spray on, this is all done on tomato plants. If you just spray on bean, that's in the top right, sorry, this color is not coming out that well on the screen, but this is the cluster here, this purple. If you just spray bean onto tomato, it looks very different than if you spray tomato selected line on tomato. But when you spray a bean and tomato together onto tomato, basically the tomato line wins. So another way of saying that if you adapt a microbiome to tomato, that adapted line will be uninvadable by another line you've adapted to bean, for example. So we did see the emergence of specificity of our lines over time on different hosts. And when we take that swapped treatment and we apply it to new hosts, in this case, we used canola, sorghum, and corn. So these are plants that weren't in the experiment at all. And then we ask, again, this host species identity effect of the lines. What we found is that the conspecific tomato line had looked really different depending on which host you sprayed it on. So it looked different on sorghum than on canola or tomato, but the swapped lines lost that host identity effect again. So they kind of assemble fairly similarly no matter who you spray them on, is the takeaway here. And the final part of this puzzle is just that if we look at some network analyses within these microbiomes, we found that relative to that host swapping line, the conspecific lines where they were maintained on a single host were able to develop more microbial interactions, more edges, and also had less clustering than the host swapped lines. So if you're interested in this idea of either microbiome passaging or microbiome selection for generalism, this paper is just about to come out, I think, next week in Cell Hosted Microbe. We're very excited about it. And also just generally excited about this type of microbiome selection. So if you're thinking about going into this space, you're already there, please reach out. Okay, with my last minute, I'm actually, I thought I took that out. I'm going to skip that. I introduced half of this data set already, but I just want to quickly wrap it up. So basically, this is Eli, who I already introduced. He developed a 16-member synthetic community of the tomato phylosphere, where we were trying to mimic the natural breadth of diversity we find on tomato plants growing in nature, but obviously do it in a repeatable, scalable way. And when I've already shown you half of this data, which is the control plants, when we spray Phylostart just early in development onto tomato plants, then we find that the Phylostart taxa, those are the dark colors here, become the dominant members of the microbiome and reach appreciable numbers on leaves relative to plants that don't get sprayed with any microbial inoculum. And this difference, the fact that control plants were so poorly colonized in a greenhouse setting, allowed us to ask for the first time whether the phylosphere microbiome has any impact on tomato yield or plant growth. And I'm just going to show you the result of one trial here. We've done this numerous times now, but if you spray Phylostart early in development and then you stop spraying, you allow the plants to grow up all the way to fruiting, what we find, and this is just across different fertilizer treatments on the right, on the top, is that the Phylostart treated plants produce more tomato fruit. And they're not smaller, they're just as large, but more of them. So they're breaking that size number trade-off. And another way of saying that is the presence of these microbes is actually acting like an additional resource for the plant to invest further in yield, or in this case, as an evolutionary biologist, in fitness. Right. Okay. And by lucky happenstance, this amazing group from South America ended up publishing this paper just before as our paper was in a year-long review cycle, published this paper where they went out and established the core microbiome of all sorts of different tomato cultivars from their natural setting. And the green here are the bacterial taxa that are in the core microbiome, and the stars are where we have family overlap with our SynCom. So we were really excited that just by chance, we actually managed to hit on most of the core microbiome that is observed in the wild. It's also disease protective, but this is a pretty well-established story at this point. The phylosphere, just like our skin microbiome, plays a really important role in preventing pathogen establishment. And this is true on leaves as well. Okay, perfect, thank you. And so just to wrap up, I would say that, there's still a lot to learn in this space, but it's really fun to think, kind of go back and forth and think about what's happening under natural circumstances or historically, and what we've changed recently under anthropogenic change in particular. So it's a wonderful space to be in this rapidly evolving field. And in my last 1 minute, Sue wonderfully introduced this, but I just want to say, so this is the Bay Area RAMPS program in microbiome sciences. It is a network that UCSF is involved in, as well as San Francisco State, Cal State East Bay, Berkeley, and Berkeley Lab. And this program is funded from NSF for the next 3 years. We hope it will go longer than that. We get renewed if they renew. But basically, the idea behind this is that we are focusing on, it's a post-bac training program. So 1 year paid full-time research. Actually, I have that on the next page. And in network labs in microbiome sciences. And we're specifically focusing on students who were community college transfer students or had other major hurdles in their way so that they were not able to establish and get meaningful research in microbiome sciences during their undergraduate career. But we hope to get into the microbiome workforce. And so we're really excited about it. We're just we have an amazing first cohort, some of whom are here and online. And we are preparing for the next round of recruitment. And the way this will happen is first, we're recruiting for new network mentor labs. So if you are a graduate student or postdoc who would be interested in mentoring a future RAMP scholar, please approach your PI and tell them to reach out to us, because we are very open to new labs joining the network and hosting future RAMP scholars. Okay, and I can take questions about this as well as the science now. I think I thanked everyone as I went, but awesome group, and thanks to them for all the hard work. Thank you to you for the invitation. Oh, I don't know where the mic is. Out there. There was one up top right. Yeah.
But online, yeah.
I thought that was a great talk. I was just wondering if you have a sense of how much this might scale in wild environments where there's a lot more potential neighbor actors, and specifically whether you think it's the selective pressures are so extreme that those communities that are naturally in the wild are already kind of in this staboid state, or is it more kind of a disruptive type of environment?
Yeah, it's a great question. And so I can answer it with just a specific focus on that phylostart, the SynCom, because in the paper, we included a field trial. So we did try where, what I didn't know when I started working on tomato is that tomato that's grown agriculturally is always started in the greenhouse. So I got excited about this because this is an opportunity to potentially seed a microbiome before you outplant them into the field. So we did that, and it's the same trial I showed you here. Half the plants stayed in the greenhouse and half were planted out. And in that case, when we got to fruiting, we saw no effect whatsoever. And my takeaway there is that this really is, as you said, it's specific, if you get a microbiome, that's great. But in a greenhouse setting, you are not acquiring a large enough or diverse enough microbiome. And so really thinking about redressing where there might be problems in establishments, not just thinking about dumping microbes out there in a setting where there's already diverse microbial. But thanks for the question.
Thank you. Excellent talk. I barely know enough about the plant microbiome to be considered a seed in the space. So please forgive the question. But I found it very interesting how phyllostart could increase yield without compromising size. I'm aware that, for instance, the tomato, which you focused on, genomics papers have shown that over time, the quality of tomatoes, it's not just consumers that have been like, it's not just consumer disgruntlement, and and nostalgia. It's the tomatoes, the volatile compounds have actually been decreasing, which has made the tomatoes taste worse over time, because yield has been prioritized in terms of just tomato amount. Do you see any potential for phyllostart in restoring like volatile organic compounds and other taste contributing components to the tomato, or other foods? Like, do you see the plant microbiome as having a role in that space?
Yeah, great question. And I mean, the short answer is yes, absolutely, I do. The longer answer is we have only anecdotal evidence at this point that sugar content and lycopene content does seem to be higher in our phyllostart tomatoes, but we're testing that explicitly now. And I've not done the sort of taste test, but I would really love to recruit enough people to the lab one day. I mean, unfortunately, moneymaker tomatoes are horrible tasting. They are canning tomatoes. So it would be a bad taste test anyway, but maybe if we move to a nicer tasting tomato, we could do that. But in terms of the quality part of your question, like the actual, content of lycopene and other things we care about, absolutely. I think that that's a huge and unexplored space at the moment. Thanks.
One more.
So I was really intrigued by your absolute abundance measurements and the control tomatoes. I guess being a newbie in this field, I would have imagined that if any bacteria got on the leaf, they would just grow up, as much as they could. So do you have theories for sort of why they're stuck at low abundances there?
Yeah, in terms of the control ones? Yeah. I mean, to be honest, I think, remember that when we're sampling here, we're sampling at the whole phylosphere level. So we take we shake everything off the above ground. And so the low abundance, each leaf is going to be its own environment. And what we know from, for example, Johan Laveau at UC Davis and others have shown really nicely, and Steve Lindau, that leaves have a carrying capacity, absolutely. And so you're asking why then wouldn't those little guys there reach carrying capacity? And my hypothesis is that they are, but they're on very few leaves, for example. So most leaves are not, and it's really not even leaves, to be honest, it's microenvironments on leaves. So little water droplets and other areas near stomata where bacteria are growing. My guess is that the vast majority of those open habitats are empty, unless you apply microbes there. Yeah. Thank you.
We're gonna break for coffee now. Just a quick housekeeping announcement. Those of us who are presenting posters, please come and meet some of the BCMM representatives out in the hallway, and then we'll resume the next session by
11:05.
11:05.
Give everybody a full break. Yeah.
All right. Thank you.
Yeah. Watch the time. Yeah. Little trouble judging time on this, so like.
I'll give you like
So I have, so you have a 30-minute session. I have at 25 minutes, I'll give you a 5 minute, 3 minute. Yeah.
and then that'll be that's the whole thing. Stop the question.
Yeah. We've been running about 32 minutes. All right, we're gonna get started.
Okay, welcome back, everyone. Please take a seat. Okay, so it's my pleasure to introduce this session. For those of you that don't know me, I'm Peter Turnbaugh. I work here on the Parnassus campus, and our lab studies how the gut microbiome affects nutrition and pharmacology. And so I'm really excited for this next talk. We're going to move from talking about plants into thinking about the digestive tract, which is near and dear to my heart. We're also gonna hear in the next talk after this about some really interesting protists that live in the gut. And then we'll move to thinking more about communication between microbes, which is a really important topic. The final talk of the session. So with that, I want to turn you over to Maria, who's going to tell us all about the fermented foods we should be eating and help you kind of decide what to have for lunch today.
All right, they're all good foods. Thanks, Peter and Sue, for the invitation to be here. A little disclaimer, I'm just getting back from Greenland. Really excited opportunity to go there, studying their fermented foods. And I guess I intentionally left my title to be open-ended because I wasn't sure where I wanted this talk to go. But I'm inspired by some of our recent work on metabolism of lactic acid bacteria. So if you're really hoping to hear about food microbiomes, I'll save that for another day and just bear with me. So let's just start by asking why fermented foods. It's estimated that approximately 1/3 of our diets is comprised of fermented foods, and this is globally, maybe not us locally. We may eat very few fermented foods, or if bread would count, or alcoholic beverages, so maybe add those in, but worldwide, they're really important staples in the human diet. So what are fermented foods? Sure you know, and just to reiterate here, these are foods and beverages that are made through desired microbial growth and enzymatic conversions of food components. And we recently revisited this definition to add clarity to, we hope, to what we're talking about here when we mean fermented foods. But ultimately, they are microbial gardens, they're microbial resources, and they're highly enriched. And these microbes are highly enriched when fermented foods are being made. Just as we think about, we're less than 1% human when we add up all of the microbes in their genes and on our bodies. This beautiful fermenting pepper dish, I estimate 50–80% microbial. But if you have, for example, this or a cup of yogurt, you're eating more bacteria than people ever lived on Earth. So they're not insignificant, reaching about 10 to the 8, 10 to the 9 cells per gram. And what we know about fermented foods is that they're really different from the starting ingredients. And these microbes impart desirable sensory qualities, extend the shelf life of the foods, increase the safety of the foods, and anecdotally, right, used to be that drinking beer was safer than drinking water, and still is in many places across the world. And actually, maybe why we think of fermented foods is through health promotion. And I want to touch on this because it's still striking to me that for now 6 years running, a national survey of dietitians rank fermented foods as the #1 superfood. That's interesting compared to what I would say, superfoods, salmon, kale, blueberries, nuts. For some reason, fermented foods have this patina as being healthy. And in particular, thought to promote gut health. And we ourselves have taken a look at this in a national survey that we're just wrapping up, had about 750 respondents. And we wanted to gauge what people know about fermented foods. Do they know what they are, what kind of foods are fermented, and how they can be healthy for us. And you can see here in the frequency of responses for what is perception is that fermented foods improve our gut microbiome, improve our digestive benefits, or probiotic, above their nutritional value. So I think this is interesting and something to follow, especially since there are very few clinical trials and observational studies on fermented foods collectively or individually. But I like to think about how microbial growth in our foods can change them so that they could be healthier. And I like to think about it in 3 different ways. So microbes will transform the food ingredients, improving digestibility, removing toxic substances. synthesize new compounds like vitamins or antimicrobials like bacteriocins, and even increase the number of living microbes like we would have if we had fresh sauerkraut, kombucha, kefir, yogurt, etc. So not all foods, but. So I'm interested in working in these different areas of research, but I want to go to maybe one reason why the public, consumers, we ourselves may think that fermented foods are good for us. And that goes back to one of the most dominant group of microbes in fermented foods, and that would be the lactic acid bacteria. So if you're not familiar with this group of microbes, they're gram-positive in the Phyllota Firmicutes phylum. And you see the definition here, and that we know are they're kind of specially grouped together. They are phylogenetically related, but they do make lactic acid as one of the main end products of energy conservation metabolism. And you may recognize some of these genera names here. I want to point out that they're pretty diverse group of microbes, and until recently, there were about 260 species of lactobacillus, with new species being discovered every year. And so through genome sequencing, ANI, and kind of regrouping of these microbes, we now have 25 genera of lactobacilli, where there used to be one. And so some of these names are complicated, like Lactobacillus plantarum used to be Lactobacillus plantarum. So I'm going to be talking about this particular microbe in particular too. So why lactic acid bacteria is being healthy, being dominant? Well, we find these microbes in our own microbiomes in the vaginal tract, where they're highly abundant in healthy vaginal tracts. And you can see here the sort of associative studies where we see lactobacilli coming out time and time again through untargeted approaches like metagenomics and 16S, and as well as through mechanistic studies in animals. And I also point out here probiotic work as well. So mostly associated with health, sometimes with disease. So they're familiar names. We find them in our digestive tracts. And then we look at individual microbes, like my favorite, Lactobacillus plantarum. We see that it is active in the digestive tract, and it seems to be particularly attuned to modifying epithelial function. We see this in human duodenal biopsies, where administration of plantarum increased the localization of ZO1 in the tight junctions. We see this in mice, where giving plantarum, again, we see increased localization of tight junction proteins. And then I'm working with Satya Dendekar at UC Davis, who studies HIV and mucosal immunology. And we're starting to tease apart exactly how plantarum is able to induce epithelial repair shortly after SIV infectious and Rees-Cox, and then even in chronically infected animals. So that's all I'm really going to go into when thinking about the health benefits of fermented foods, and why I think there's this connection between what we eat and thinking about beneficial microbes like lactobacilli and the fermented food themselves. What I want to kind of go to is actually fermented foods are really diverse. And we performed a community science project, again, during the pandemic, when we're able to be in the lab and ask for donations, people making their own sauerkraut, kimchi, fermented cucumbers, chili peppers, whatever they were making, fruit and vegetables at home, and surveyed them by 16S sequencing. And you can see just a diversity of just, it's a very high level, right? But quite a few of the plant-associated bacteria still there, but mainly dominated otherwise by lactic acid bacteria. When we look at our own ferments at home, maybe if you ferment, you'll see that your different batches are quite different from one another. Even if we have a cheese where we add a starter culture, mostly Lactococcus, that we would expect because we had a lot of them, but underneath that, there is also a lot of variation. Another example here is in fermented olives we made at home, and these are just using the same recipe, same day, same olives, very different appearance. And lastly, one metric of a good fermentation is the capacity of these bacteria to reduce the oxidation reduction potential to make their environment more anaerobic is one reason. And you can see there's a lot of variation in the capacity of microbes to do this. So this brings me to metabolism. When we think about lactic acid bacteria, you probably think of lactic acid, and it's probably pretty simple, but it isn't, of course. So there's two kind of pathways that have evolved in lactic acid bacteria for energy conservation. One using glycolysis here, and where they reduce pyruvate to lactate, and that's released from the cell. And you could see they need to do this because of reduced NAD in the cell. They need to regenerate in order to continue to grow. And the heterofermented pathway, these microbes lost the aldolase, and instead they use phosphokedolase. And this results in sort of a mixed acid fermentation where you have lactate as well as usually acetate and some ethanol. This is a really simple way of looking at it. If we have oxygen present or not, a different kind of carbon sources available, even homofermentative LAB have diverse pathways and diverse end products that they make as a result of their need to maintain redox status in the cell, and also potentially get a little bit more ATP. And the same goes for the heterofermentive microbes as well. Interestingly, about 20 years ago, it was found that some of these bacteria can also respire. They don't possess or cannot make a complete electron transport chain, but they're able to respire when oxygen is present, typically, heme and metaquinone is available. But there's even more to this. So this is where I'm going in thinking about what we don't know about fermented foods. And it turns out we don't know a lot. So when we think of extracellular electron transfer, or the capacity to move electrons outside the cell, we typically would think of soil microbes like Shigella or Geobacter. These are anaerobic microbes. They find them in sludge or in rhizosphere. And they're gram-negative bacteria. And so this is how we've understood this electrogenic capacity of microbes. But can other microbes do this as well? So to explore this, we took our favorite microbe, L. plantarum, and we gave it a carbon source. We gave it quinone, which we think could be an electron shuttle. We know that plantarum cannot make its own quinones. In this case, we gave it DHNA. And then a very simple assay as we look for the capacity of this microbe to reduce extracellular iron, using ferrozine to detect that iron reduction. So the purple color here shows that iron is being reduced. And so to see this, we perform this assay with these components, with the live microbes, glucose and DHNA. And what we found is you need to have all three. The plantarum needs to be alive, it needs to be quinone, and the carbon source available. We also measured this by quantum amperometry, so by electrical cells. And with the polarized anode, we could see that plantarum is able to make electricity when glucose or carbon source is present, as well as quinone is there. And it became quite exciting because we found that plantarum is actually able to make even more current than Shigella, which is the model organism for mediated T. So this resulted in a really nice collaboration between myself and Eric Stevens, who was a graduate student in my lab, and Caroline Adjo Franklin, now at Rice University, and her postdoc, Sarah
George Sands, and Sam Light, who's now at the University of Chicago. So just for example, Siliang, who's in Carolyn's lab, showed that plantarum, these microbes who never thought could make electricity. And I'm going to try to play this video here, connecting an LED between the two chambers. See that it's able to make light. So maybe for your next fermented foods, get some energy generation. So we went on to kind of explore, is this really a unique metabolism, or how does this work in L. plantarum? We do know that it can use flavins for EET, but also that you need to have this quinone present. So plantarum doesn't make flavins, doesn't make quinones, it's really reliant on the environment. And so I'm going to come back to this concept later on. This particular organism can respire anaerobically. We deleted the nitrate reductase genes in plantarum and found that this metabolism is not relying on anaerobic respiration or proton motive force. We went on to say, well, what particular genes are going to be involved in this EET metabolism. And for that, we borrowed work from Sam Light, who showed that Listeria monocytogenes can also perform EET and possesses this EET or fleet locus. And looking at about 2,000 lactobacilli or LAB genomes, we found that about 11 out of 38 genera possess this complete fleet pathway. And 19 out of 94 lactobacilli species contain it. And so we wanted to kind of narrow down which genes are going to be most important for this EET metabolism. And so we took isolates from these different genera and put them into our iron reduction assay. And what we found was that two genes are essential for this metabolism in LAD, one being an type 2 NADH dehydrogenase, and the other being a gene that reputedly encodes a flavin binding extracellular lipoprotein. So we found that these microbes need to have both of these genes to perform iron reduction. You'll also notice here that there were a couple of plantarum strains in our collection that didn't reduce iron, which was counter to what we predicted when we looked at all the plantarum genomes. They all were supposed to reduce iron, but these two strains have transposon right there in the intergenic region between NDH2 and PPLA. I wanted to make sure that again, these genes are important. So we deleted the NDH2 and PPLA genes from plantarum, and we didn't get iron reduction. Interestingly, NDH2, but not PPLA, is important for making current. This could be relevant in fermented foods because, as I pointed out, we have this ORP as a variable phenotype in fermented foods. We know that removing oxygen is one factor, but it is not the only. And the progression of any fermented food will depend on it. And so it was interesting to see that our two mutants, NDH2 and PPLA, didn't result in the same low ORP values as the wild type strain. So what's in it for the microbe? To that, we turn to our electrochemistry, and the setup is here with the electrode on, or ET on, and open circuit where the electrode is off. Interestingly, we found that EET does confer an advantage. We see within the first 24 hours a significant increase in the cell weight and cell numbers. And what we see is that EET imparts a capacity for plantarum to grow early on in the fermentation. So we see a significant increase in cell numbers early during the fermentation. You can see later on the open circuit non-EET condition, these microbes still growing, just so that it takes more time. We also looked to see what was happening inside the cell. And under EET conditions, here we see for about 4.5-fold higher levels of ATP and 8-fold higher levels of NAD to NADH, so more oxidized state in the cell that are actually resembling what we see in respiration. What happens to the metabolism, the fermentation pathway? We had several predictions. Well, first of all, what we saw immediately, though, is under these closed circuit conditions where ET was possible, we see a dramatic reduction in pH. And that was basically due to the enrichment of lactic acid in the medium. And what we see is an overall increase in flux through the fermentation pathway. And so we get, instead of less lactic acid, or what we would see in respiration, we see increasingly more. So under open circuit conditions, we hypothesized this pathway, and then we have this increased flux through fermentation. Is this happening in real life? That's something we're exploring now. So to start with, we just took some kale, and this is what happens to kale juice when you autoclave it. Doesn't look so good anymore. But we wanted to control the other microbes for this first experiment. We added in quinone, which we found that we needed to do, and we see current. So even in a complex matrix like a kale juice, we get EET activity. And we also were able to recapitulate what we saw in culture media with a significantly reduced pH due to the production of more metabolites, organic acids during growth. Just a few more minutes, I think, that I wanted to give you an idea of where we're going with this. As I mentioned, Plantarum is unable to make its own quinones. It lacks the upstream part of quinone synthesis, but it can potentially make metaquinone if DHNA is present. We wanted to know, well, if this capacity for EET was specific to DHNA, and so this is what we saw when we add DHNA to the culture medium. We don't always see iron reduction, and so for certain quinones we do, like ACNQ, which is a metabolite of Lactococcus lactis, which does make quinones. And so this brings me to the complexity of these food fermentations. We're going to have different species of LAB growing together, as we would in a sauerkraut, where it starts off with Leuconostoc mesenterides, a heterofermentive LAB that can make its own quinones, and then Plantarum comes in and starts growing. And then in any cheese fermentation, you're also going to have Plantarum together there with lactus. And so we were thinking, maybe what's happening in these fermented foods is that there's this cross-feeding in quinones and metabolites. And so we took our quinone-producing LAB, super named after their growth, and incubated it with Plantarum, wild type, and the NDH2 deletion mutant, just to show that these microbes do make quinones. We measured them in collaboration with Emily Mevers at Virginia Tech. You can see those are intracellular metaquinones. We also received MEN-C deletion mutant, unable to make quinones of Lactococcus lactis. And lo and behold, when we incubate Plantarum with the supernatant from these strains, we see iron reduction when the wild type Plantarum strain is used, but not with the NDH2 deletion mutant. This is with Leuconostoc strains, same with wild type Lactococcus, but not the deletion mutant, unable to make metaquinones. So suggesting that there's this cross-feeding between these microbes in a food fermentation. What happens when we put them together? We see that just as we saw when we grow plantarum individually, performing EET, accelerated reduction in pH, and for the leuconostoc co-cultured cells, we see a blip of an increase in growth in plantarum. We don't see that with lactus, so we're currently sorting that out. So now we're seeing EET everywhere. It's we see it in papers, of course, in the mouth, the lungs, the gastrointestinal tract. In our own work, we found that NDH2 and PPAL are highly upregulated in the small intestine of rhesus macaques. We see it happening, of course, in the rhizosphere and in foods, and we invite you to also look for EET in your microbes. So in summary, fermented foods are really exciting. They're great sources of live microbes in the diet and microbially derived bioactive compounds. This type of metabolism is pretty new. It's a hybrid metabolism. It's not respiration. I break that down here. We think that it's this hybrid, but ultimately, they're really still underexplored. We talked about intraspecific diversity or interspecific diversity of these bacteria. There's a lot to be uncovered, but a lot of biotechnological potential and capacities, I think, ultimately to improve human health. I want to give a shout out to Eric Stevens, who did all the EET work, Lei Wei and Mons van Beek, who have been carrying forward with this, and our community science research, and my collaborators, Carolyn, Sarah, Sam, Emily, and then at UC Davis, Aaron DiCaprio, Melanie Hamlin, Andre Knudsen, and the funders, and I'll end here with your questions.
I thought that was a really exciting talk, and I just wonder how many other members of the gut microbiome in particular you think this mechanism might extrapolate to. You focused on these lactobacillus species, but whether you think this is actively ongoing in just firmicutes, or if it's spreading to different phyla or even
thanks for asking that question. I think redox status in the intestine is something that we have to be paying attention to. David Lawrence had published a nice paper a few years ago looking at redox balance during antibiotic treatment. Just to highlight, Faecalibacterium here seems to also do, EET. You can see it there, but colorful microbes. So yeah, I think what we need to be looking for is the potential for this metabolism, how this would be possible in the digestive tract, what are the electron acceptors. Also some really interesting work suggesting that that there could be immune cell recruitment out there, but depending on the redox state of that microbiome.
Hello. Sorry for my banal question, but we can be sure that diet-derived microbes could survive after stomach digestion.
Yeah, you're asking, do lactobacilli survive?
Yes, and all food-derived microbes.
I think I think also. Yes, there's been shown in many ways. We know that they're metabolically active. At least the microbes that I work with are metabolically active in the small intestine. Not all the microbes are going to survive. If you look at fecal samples or transcriptomes and fecal samples, you do see fermented foods and fungi, bacteria, that at least the DNA or RNA is there. And so, yes. And it's, of course, it's going to vary depending on the microbe and the context that it's eaten in.
So there are some tests to verify this. Yes.
Yeah. You could look at the tag strain or strain diversity, follow individual strains in the digestive tract. Yeah.
Yeah, thanks so much.
Go ahead and down the line. Oh, you just need the one.
Oh, I was gonna do this first slide of this.
Oh,
Or,
yeah, sure.
or is there another? Whatever.
Does that work?
Well, this isn't hooked up to anything. That's the.
Oh, no.
Okay.
Okay, so next up we have Michael Hatwood, who joins us from Stanford, and he's going to be telling us a lot more about the gut and some interesting types of cells, both protists and host. So,
Thanks, Peter. Good morning, everybody. Thank you for the invitation to speak. So, yeah, as Peter mentioned, I'm going to be talking to you about two interesting types of cells. The beginning one actually is going to sort of reflect the lab's interest in host biology, right? And so actually how I became interested in the microbiome was an interest in trying to understand how the gut microbes were interacting with the epithelium. Those of you probably may or may not be familiar with epithelium of a person, an adult person, is about the surface area of double tennis court, right? So this is an incredibly broad, expansive area of microbial host interaction. And when I began my postdoc, this was kind of the landscape of the gut epithelium in the small intestine. In other words, there was considered about 4 main differentiated cell types, all arising from the same common stem cell. And many of these have known functions with the microbiome. The aspect that actually caught my attention is that it wasn't just 4 cell types, there was actually 5. And at the time, a few pioneering souls had pointed out that there was this cell called the tuft cell, so named because of this prominent tuft of microvilli you can see poking up above the brush border here, and a couple of other really striking morphological features like a very prominent cytoskeleton, et cetera. And this was not a new cell type. This has been first described in the 1950s. I now know from my colleagues who read German, possibly the 1920s, and that it's also found quite widely in most vertebrates, including our usual animal models, people, et cetera. All have cells that morphologically look like this. But tuft cells were quite rare, right? Estimates suggested they were about 0.3% of the gut epithelium, making them really a very, very tiny fraction of that vast surface area. But the element about them that really sort of garnered my attention and sort of convinced me to try to convince my PI at the time to actually work on them was this idea that these were taste cells, right? So what I mean by that is they express some of the same core taste machinery that you have in your tongue, and even in fact some of the same taste receptors they were expressing and they were using to, quote, taste things in the gut. And what I found particularly intriguing about that is a couple of studies in the upper respiratory tract had shown that the sense of taste may be involved in not just food preference, but in microbial detection, right? And so I was wondering, could these taste cells be involved in tasting some factors that microbes were making? And so the very first thing that we did is just, hey, this is a weird cell type. No one was really studying it at the time. Can we even see them in our mice? And so what you can see is tuft cells in green, which is staying in the small intestine. Those are the little villi fingers that you can see. And when we quantified how abundant they were, we saw they were about 7%, right? And this is in stark contrast to that 0.3% that I mentioned. This is sort of, they were supposed to be quite rare. We weren't really seeing them to be all that rare in our mice. Now, these mice were at Harvard at the time. We had sourced these from Jackson. And so when we got the same strain of mice from Jackson, but hadn't lived with us for a couple of years, we saw that tuft cells were actually quite few, below 1%, right? So this led us to think, okay, there's possibly some environmental difference. Could it be dietary? Could it be something about our husbandry practices? Obviously, I wouldn't be here if it wasn't microbial. And so we took the fecal contents from our mice, we fed it to the Jackson mice, we waited a few weeks, And then we saw that basically we could phenocopy everything, right? So this is a ton of tough cells. It was some microbe that was present with us that was driving this. And so at the time, again, I was coming from kind of a more of a host perspective. I was thinking, great, this is bugs. We're going to do 16S sequencing. We're going to find out the bacteria that are driving this response, because in my mind, bacteria were the sum total of the microbiome. And we found a bunch of differences, and I got nowhere, right? So this is like 6 months of like absolutely doing nothing productive. But at the same time, I just started looking at the tissues for other reasons. And this is just simple H&E. This is sort of your bread and butter pathology technique. And what we noticed was this huge accumulation of rather large cells. And if you zoom in on them, right, and you can see kind of what we anticipate are probably the bacteria here. There are these large cells. They look like they had nuclei. When we purified them, we looked at them by scanning electron microscopy. This is what we saw. These were tritrichomonads. We could verify this molecularly, but you can see kind of what they look like, right? And so this was pretty intriguing to me at the time. We looked to see what did we know about these, and they were considered to be normal members of the rodent microbiota, but not particularly important, right? People weren't looking for them. They didn't cause disease. So, right, these aren't Giardia, these aren't Cryptosporidia, but these are just considered normal. On the other hand, to my delight, I was able to isolate them, introduce them into naive mice, and then we saw this big expansion of tough cells, right? So what I had going on here, trying to understand host microbial interaction, I think was a certain degree of maybe sort of poetic symmetry, right? I was studying this host cell that no one really knew about or cared about, and I discovered a microbe that really no one knew about or cared about that actually drove that response, right? But it turned out these tritrichomonads ended up being quite useful, right? And so work from myself and my group and other folks, including Rich Loxley here at UCSF, went on to show that tough cells are really key innate, or I won't call them innate, but they're an epithelial cell, but they're a key sensory cell at barrier surfaces like the gut that sense the presence of different microbes, including these tritrichomonads, also intestinal helminths, and these convey the presence of these organisms to the underlying immune system, principally through effectors like IL-25, signaling to immune cells like group 2 innate lymphocytes. And these lymphocytes signal back to the epithelium. They give rise to that expansion of tough cells that I first showed you, right, which just sort of really caught our eye, and elaborate all the other aspects of type 2 immunity. So this is a really series of interesting stories. We continue to work on sort of the host effects from this. But one of the things that we realized at the time is that really all the biology that we understood was down here on the host side, and we really knew not much about these trichomonads at all. We knew from work that like Rich and Jakob von Moltke had done that succinate was a key metabolite they were producing, and that's what tough cells were sensing. But we didn't really know much about them. We didn't know what were the different species, how diverse are they, were different groups working with different strains, different species. Could there be other effects other than just stimulating tough cells? We didn't have any genomes for these organisms. That was one of the reasons why we couldn't check for these things. We didn't know what their role was within the microbiome, right? Was it important that there could be different microbiome settings in which these organisms were operating? How does diet affect? All of these things we didn't know. We couldn't do basic stuff. We couldn't culture them, right? So like we really didn't have much to go with. But what we wagered was that there probably was more than what we could appreciate because we lacked all of these tools. And so when I moved from the East Coast for my postdoc to start my lab at Stanford, everyone warns you, hey, you're going to move your mice into a different vivarium. Your microbiome issues are going to cause problems. Your phenotypes are going to go away. To a certain extent, they were right. But one of the other things that we hoped to do is we said, okay, maybe we can look for our West Coast mice and we can find some new species of tritrichomonads. And so this is a project that's really been spearheaded by Eli Garrick here, who's a postdoc in the lab, and Claire Meckler, who's a technician. Both of them are absolutely amazing, and I'm really going to be talking about their work. And so one of the things that we first wanted to do was start checking all of our neighbors' mice. So like I move in and I'm like, hi, nice to meet you. I'm Michael. I'm new here. Can I have some of your mouse poop? And they gave it to us, and we found nothing. And so I thought maybe our coverage was a little low. So I convinced the veterinary staff to let us look at the sentinel mice, right, where they were looking for specific pathogen-free, thinking this would give us broader coverage, and yet we still found absolutely nothing. So this is something that when I give this talk at Stanford, people are pretty happy about, but I was not, right? But Eli actually had the good idea, well, what about looking at our own mice? What about turning attention onto us? And so he started really looking in a more granular level at our own cages. And what he found is actually our own mice contained multiple types of these tritrichomonads. And so we could see this both molecularly and by microscopy, but what I'm showing you is just flow cytometry, a fact representation of that. And you can see here, there's two different size protists, right? And so what we asked is are these two different morphotypes of the same species, or might these actually be two different types of protists? And so we flowed them using sort of this size differential, introduced the large protist into a naive mouse, introduced the small protist into a naive mouse, and what we observed was continuity, right? So if you give a mouse this large protist, you only have large protists. You give the small protist, you only have small protists, suggesting that these weren't going back and forth in between these different sizes. Of course, that's not particularly good proof. These are different species. And so we actually, in collaboration with Amibad's group, we sequenced the genomes for both these organisms. I'll just sort of show you the very top line number here in terms of the 158 megabases versus 64. These are unambiguously different species. You can see them in terms of the phylogenetic arrangement here. These are all parabacillid species here. They are still closely related, however. So the first question that I wanted to know, right, being interested in the host immune system is, do these things drive tough cell responses? Do these things drive type 2 immunity? Because at this point, every single trichocomona that anyone had published on does do that same thing, right? So this is what our expectation was. And what we found is, yes, of course, as our control, T. musculus, which is the original trichomonad that I had been studying as a postdoc, drives this response, but T. casperi, which is what Eli named it for reasons I won't get into, it does not, right? We didn't see a tough cell response. We didn't see, and this is a CD4 Th2 response. In fact, we saw a slight reduction in this. So it wasn't driving the same immune response. We then wondered, well, is this basically immunologically inert? Is it just failing to drive this type 2 response, or is it doing something different? And it turns out actually it is not immunologically inert. So this is all in the small intestine. It drives a Th1 and Th17 immune response, right? And so that's indicative of, again, CD4 T cells. We're just looking at some canonical cytokines, interferon gamma, IL-17A, right? So these are basically, they're colonizing the same regions of the gut, but they're driving disparate immune responses in these regions. And so then we turned back to, okay, what do we know about these protist biology? What could explain some of this? And so Maria already introduced a little bit about microbial metabolism. This is a highly simplistic schema. Basically, as she mentioned, if you need to oxidize NADH into NAD+, T-mu does this, and it generates a bunch of succinate and excretes that succinate, and that's what triggers tough cells and triggers that type 2 immunity. So we asked, well, is this happening with T. casperi, right? And again, we just confirmed this by mass spec. If you look at mice that are colonized with T. musculus, you look in the cecum, there's a ton of succinate, and there's nothing in T. casperi, suggesting that perhaps these guys are not making, or at least not excreting succinate. And so again, we had the genomes, we assembled the metabolic pathways that would be responsible for generating this NAD+. And so in blue, you see the genes here. They're encoded by T-mu, so fumarate reductase, converting fumarate into succinate and regenerating NAD+. We could see in T. casperi that it actually encoded both pathways. It also encoded lactate dehydrogenase. And so what we assumed here was that it wasn't producing succinate because it was regenerating NAD+ through generating lactate, right? This is what we thought we figured out. Turns out, and I just want to mention also, we did this all in collaboration with Sumaya, who is joining the center here and launching her own lab. This is just showing you a snapshot of some of the metabolomes we found with her. She's incredible. We looked at lactate, and to our dismay, basically, we saw hardly anything. And this actually was somewhat predicted because we looked at RNA-seq, and we could see that T. casperi was expressing lactate dehydrogenase, but at much lower levels than fumarate reductase. And so when we actually took the protists themselves, we didn't look at sort of the extracellular metabolite environment. We took the protists and we cracked them open and we looked what was inside them. We actually saw they expressed tons of succinate. And so what we hypothesized is going on is they're regenerating NAD+, also using fumarate reductase, generating succinate, but instead of excreting this succinate, they're converting it to something else, and then that is what is being excreted. And so we've done a bunch of metabolomics, again, with Sumaya and others. We have some ideas, but we don't yet know that answer, right? So the next question we wanted to know is, what about metabolic input, right? I've been spending a lot of time thinking about what is their metabolic output. And the presumption had been that these trichomonads, like many microbes in the gut, are fermentative organisms that take advantage of all the fiber in our diet, right? But answer is we didn't really formally know this. And so we started to try to use defined diets using fermentable fibers like inulin or non-fermentable fibers like cellulose. And so what we did first with T. musculus is we had the complex chow, which is normal mouse chow, and we had defined diet. And so CI stands for cellulose and inulin. We had no fiber, or we just had cellulose, or we just had inulin. And basically what you can see is when you start removing fermentable fibers, TMU does not do well. It drops precipitously in numbers, right? Suggesting that it needs this dietary fiber to be available. And this is actually what one would predict, right? When we looked at T. casperi, we found it didn't care. You could totally take fiber away from the diet and it persisted just fine, right? So this was like a huge difference already between these guys. And so we thought, okay, what might it be doing, right? What is it possibly eating? And so again, we didn't know much about their biology, but we figured, okay, we probably know a little bit more about the bacterial microbiome. And one of the cool things about these protists is that they don't seem to be super susceptible to colonization resistance, right? So you can take an intact microbiome that's really established, and oftentimes that can be difficult for new microbes to come into that. These guys don't really seem to have a problem. You can colonize mice, once with this, use 1,000 of these trichomonads, and they take very well, right? So they're very good at establishing themselves. And what we figured is that it was unlikely that they were accessing a new niche that bacteria hadn't figured out. That's what our hypothesis was. And so we thought perhaps we could get a sense of what they were eating by looking at what impacts they were having on the bacterial microbiome. And so we did 16S, and we looked at some of the microbes that TMU and T. casperi were displacing, and I'll just kind of highlight a couple that we thought were interesting. And so if you look here in sort of this turquoise here, you can see bifidobacteria and Terecibacter, organisms associated with fermentation of dietary fiber. These were decreased, and this is again just across sort of different sections of the gut for those of you who may not be familiar with that anatomy. One of the things that we thought was interesting about T. casperi, which was more of the mystery, is that we saw that it was driving Akkermansia mucinophilia to extinction, right? And so as the name would imply, Akkermansia mucinophilia, this is a bacteria that loves to eat mucus. And so when you think about mucus, although there is obviously a protein component, it's about 80% carbohydrates. So this is a potentially really rich source of sugars if you have the enzymes to cleave them off. And in some bugs like Akkermansia, they do. And so we then looked at the RNA-seq databases of these protists, and we asked, are these expressing the enzymes that are necessary to cleave off the sugars to actually access this nutritional source? And so when we looked at T. casperi, we looked at these glycoside hydrolases that were predicted to be host active, right? So non-plant active hydrolases. And to our delight, they expressed a ton of them, including all of the sort of necessary glycoside hydrolases that you would need to liberate these sugars off of the mucus. So this was consistent with our hypothesis they were eating mucus. To our dismay, however, T. musculus, albeit at a lower level, also expressed these same genes. So that was kind of a rub. Why was that happening? And so one of the things that we did is we sort of turned back to our data where we looked at cellulose, right? So this is a non-fermentable fiber. When we look in TMU in a complex diet, right, they're distributed all over the place. This is the sort of the host layer, the mucus layer. This is the host tissue. When we looked at it with cellulose, they way less of them, but there was a few, and the few that were remaining were hanging out here in the mucus layer, right? And so one of the things that has caused this thing is perhaps they're just in a really sort of nutritionally tough environment. They're able to hang on through fermenting mucus. And so one of the cool things that Eli did, and this is, I told him was the most exciting, boring piece of data he generated, which is he actually figured out how to culture these organisms. This is something I'd worked on for a couple of years as a postdoc. I couldn't figure out. He worked on for a couple of years. I was like, crap, we're never going to figure this out. But we've been able to grow these things now for months continuously. And this is great for a number of reasons. One of which is that we can start looking at what's their preferred carbohydrate substrates. And so we're actually able to add in, maltose, which they love to eat. We added in mucus as the sole carbohydrate substrate for TMU, and they grow great, right? So they love it, right? So why are they doing so poorly in vivo, right? If they're able to survive on mucus. And of course, one explanation is, well, we're missing the microbes here. And so we went and we decided to broadly deplete bacteria by treating with, Vanc, Neo, and ampicillin, and we basically saw that could sort of rescue their levels when we were giving them just cellulose. In other words, they didn't have any dietary fiber to eat, and they did just fine when we killed the bugs. So to get a little sense of who it might be, we sort of parsed these antibiotics into single treatments, and we found that really this effect was completely driven by ampicillin. Neo and Vanc were unnecessary. And so we did 16S. We looked at, okay, what were some of the bugs that were being changed in these treatments? And I'm just going to I'll quickly show you also what it looks like, right? So if you give ampicillin, you rescue their numbers, but if you look at them, they're not distributed everywhere. You see them just piling up here along the mucus, suggesting that this is opening this nutritional niche for them. And again, when we looked at 16S, I'm just giving you sort of the take-home point. We saw that ampicillin was the only treatment amongst these antibiotics that was getting rid of the bacteria that are sort of canonically associated with eating mucus, right? So keeping with the idea that this was perhaps opening this mucus niche for them. And so the other interesting thing is that we noticed that if you gave them inulin, that would rescue them, right? If you give them inulin and cellulose, you get lots of T-mu. And this was a bit puzzling to us because we noticed that if you feed them inulin in vitro, they actually don't grow on it. It doesn't appear like they like to eat inulin. So how was it that even if you gave them only inulin, you could actually allow them to survive? And so again, if you actually look here by microscopy, again, this is a complex chow, they're distributed all over the place. In cellulose, you have very few of them, they're in the mucus. In inulin and cellulose, there's lots of them, but they're all here on the mucus layer, right? So they're moving. And what we actually believe is happening is that the inulin is not providing the trichomonads a food source, it's providing bacteria that they're competing against a food source, and it's opening up this mucus niche, allowing them to live in that. And so essentially this is work that we've just published. So it's in press now, but it's not out. And essentially this is the model that we have two organisms living on different polysaccharide sources, T. casperi being more of a mucin specialist, T. mu being more of a generalist that prefers to eat dietary fiber. It can compete against some of those bacteria like I was showing bifido, whereas T. casperi negatively impacts bacteria like Akkermansia mucinophilia. If you remove fiber, you basically, bacteria now are out competing T. mu while T. casperi is still able to maintain. And then if you add back in organisms like inulin, the fiber, as I mentioned, we think that distracts some bugs. Probably we don't know yet, but we're thinking Bacteroidetes, that then allows T. mu to access this mucin niche. And the other interesting element that I didn't really have time to get into is that they do produce succinate, but it also, using diet, can modulate the immune system. So there's some other interesting immunological findings here that I didn't get into. And so I'm just gonna finish up with my last slide. Hopefully, saying that this proteus diversity exists in mice, that this diversity is functionally meaningful in terms of its impact on host immunity, but also on the microbiome. You might be asking, well, okay, mice are cool, but what about humans, right? And so we've just started some work looking at parabacillid protists. Again, this is sort of the phyla of these tritrichomonads belong to, and people. And so in collaboration with Matt in Justin Sonnenberg's lab, we looked at parabacillid prevalence in about 1,800 human metagenomic stool samples across 29 different populations, representing different lifestyles from industrialized, transitional, and hunter-gatherers. And a couple of things that I just want to kind of show you here is that looking at the prevalence, it is much, much, much higher in non-industrialized populations than it is in industrialized. So the folks that we looked at in industrialized groups, these protists are actually quite rare. But if you look globally, they're actually not particularly rare in people. And the other thing that we found very interesting is that there had only been two gut-associated parabacillid protists in humans, Dientamoeba fragilis and Pentatrichomonas hominis. When we looked, and so we did see those, as you would imagine. For instance, we saw a lot of D. fragilis. But when you look at many of these other species other than P. hominis, you can see these asterisks by them. What that means basically is that these were all probably undescribed species that when we tried to map them and say, what are they most closely related to? We'd say, okay, they're most closely related, but they're definitely not T. musculus, that's mouse associated. It's just the closest thing we have. And so what we found is that there's a ton of diversity present in non-industrialized populations that is not present in industrialized humans, and that this probably contains many, many undescribed species in it. So hopefully we can begin to understand how that diversity may also impact human health and the human microbiota. And the very just first tiny step in that direction that we tried to do is to look at differences in lactate versus succinate production as part of that redox metabolism. Both lactate and succinate are both immunomodulatory metabolites. And so we were able to use some genomic and metabolic biochemical measurements to see a phylogenetic schism in terms of whether you're going to make lactate or whether you're going to make succinate, allowing us to make some predictions about some of the new parabacillid species that we found in humans, whether they're going to make succinate or lactate. And I just want to point out a clear caveat to those predictions, is that even in our own data up here with Caspari and mustelid, right, we know that T. caspari is generating succinate, but it's not excreting it, right? So there's some imperfections to this prediction, but it's a start, hopefully. So thanks very much. I just want to thank everyone who did the work, particularly, again, Eli and Claire and everyone else in my group. AmbiBot's group is absolutely amazing. You guys are going to be blessed to have Sumaya here. We also collaborated a lot with Patrick West, who helped with some of the genome assembly. The Sonnenberg lab has been absolutely fantastic. I showed you mostly the stuff that we did with Matt Ohm and Matt Carter, but we also did a bunch of notobiotic work as well. And so this has been just a really fun team effort, and I'm happy to answer any questions.
Yeah, that was a great talk. I was curious how much of the immune effects are direct versus like downstream of the effects on the bacteria.
Yeah. So we had that same question. So we're digging in and we're seeing that there are many other new immune effects that other than what I just showed you. But in terms of looking at the T cell response, it appears to be direct from the protist, both T mu and T caspari. So we monocolonize germ-free mice, and you can basically see, for instance, like the Th2 response in T mu. You can see the Th17 and Th1 response in T caspari. And so it appears to be direct protist to hosts. However, I think, for instance, Akkermansia is a very important microbe in terms of immune effects. So we do think there is going to be other interesting immune phenotypes that are going to arise from these trans-kingdom interactions. But yeah.
Thank you so much. Never protest the protists. Excellent talk. Though I was obsessed with protists in middle school, I am definitely not super familiar with this space. But it sounds super cool that you found a method to isolate Trichomonas and to grow it in vitro. I was wondering if you were thinking of doing this for human bacillid protist species, and also how novel that method was, and if you think it could be applicable to other protists or even microorganisms within the human gut.
Yeah, so it's a great question. So yeah, so we have Pentatrichomonas hominis. I tried in the middle of the pandemic to import D. fragilis from a hospital in Italy, and just the nature of the times in mid 2000, it got delayed, so they were dead, and we could never actually revive them. In terms of some of the undescribed species that I mentioned, so I just want to say all that analysis was on published metadata, so we didn't collect or sequence any of those samples. We would probably have to actually go and have access to those populations to actually acquire the live organisms. So there's going to be a lot of practical barriers to doing that. I would love to. I think it would be interesting, and perhaps down the road set up some collaboration with local folks in those communities. I do think this is potentially more broadly applicable to other protists. In another paper, totally unrelated to this, I was talking to Peter about this. We were looking at parabacillids in the reptile microbiome, and Eli did adapt a lot of these culture techniques to culture new reptile species. Obviously, we had to make some modifications in temperature and otherwise, but it does appear there's some universality. It's not perfect. So, for instance, we'd love to be able to culture T. caspari really well. We've made some inroads, but while we've grown T. mu, I think as long as 6 months continuously, T. caspari, it always craps out on us eventually. So yeah, there's still some nuances that we don't understand.
Hello, great presentation. Is there a possibility that your protists are actually carrying out predation on bacteria, and that's the competition that you're seeing?
Yeah. It's a great question. I mean, so obviously I frame this in the context of nutritional competition, and it may be wrong, right? It may have still led us to some of the interesting conclusions, for instance, that T-calciferide may be relying on mucus, but the fact that, for instance, it's out-competing acromanthin may not because they're competing for the same nutritional source. It's a really interesting idea. That we don't know. The other thing that we think is we've looked at other scenarios where we see the bacteria out-competing the protists, and we're also wondering whether some of that doesn't have to be mediated. Some of the kinetics of that can be very rapid, and we're not sure that that is necessarily in keeping with just a pure nutritional competition, whether it could be active killing. And then there's another project that we have in the lab trying to look at fungal protist competition, and there's some evidence that maybe fungi might have the ability to kill as well. So these are all really interesting. I don't have a good solid answer, but totally we think these are interesting questions and we're sort of looking into them. Yeah.
And sorry, I want to ask the last one.
Sure.
Have you tried to grow them together with bacteria in the laboratory, like instead of providing the substrates that you're giving?
Right. Yeah, we have. So it's tricky with some of the growth kinetics. We haven't totally optimized them well. And so that hasn't been fruitful, but I think it's a matter of optimizing it. But yeah, so we've tried, but not successfully. Yep. Well, thank you.
Okay, finally, last talk before lunch, we have Corinne Gibbs from Berkeley, who's gonna kind of continue this theme of communication, moving from, thinking about these inter-kingdom communications that we heard about in the last talk, to how kind of cells within a population communicate with each other. So I'm really excited to hear about your talk.
Thank you. Thank you, Peter, and thank you for organizers for the opportunity to talk today. My name is Corinne Gibbs, and I'm an associate professor in the Department of Plant Microbial Biology at the University of California, Berkeley. And welcome to everyone on Zoom. I will try and stay in one spot, but I tend to move. Okay. Today I'm going to tell you about bacterial communities and really how do bacteria deal with their closest neighbors. And so the image you see before you here is a microscope image of our favorite bacterium, Proteus mirabilis, where all of the membranes are dyed in blue and the surface is in black. And as you can see, these communities are jostled next to each other. They're forced and squeezed, kind of like the subways in Tokyo during rush hour. And so when these organisms are working with each other, they not only have to deal with their environment, the nutrients, the air, the rough surface, but also their neighbors. And they have to take those signals, input them, and have a response. So let's start with what you're going to take away today, in case you want to leave early for lunch. One of the things the main 4 parts I want you to take away. Organisms are impacted by their neighbors, particularly when constrained in space. I'll be talking about 2-dimensional space today, but as you know, the microbiome is in 3-dimensional space. It also has dynamics over time. Organisms do not usually control which signals they receive. As with humans, we're stuck with people sending things at us. But they can control how they respond to those signals, and that's partly how they evolve. Organisms may also use kin-specific defenses and broaden those to actually use them to defend themselves against other things. And this could be important for survival, especially in mixed communities. And finally, a lot of what I'll talk about today are protein-protein interactions. And I know right now we've talked a lot about sequence data and things like that, but I want you to go all the way down to within a cell. At the end of this day, with signal transduction, we're talking about how one protein or one signal is interacting with another protein and another signal. And it's the output of those interactions that can drive behavioral responses. Okay, so with that, before I go any further, we're talking about communities. And so I want to first acknowledge that the work I'll talk about today was done both in my lab at Harvard University when I was there, as well as UC Berkeley. Harvard sits on the traditional and ancestral lands of the Massachusetts, who are the original inhabitants of what is now known as Boston and Cambridge. Furthermore, UC Berkeley itself resides in the territory of the Wichin. And this is the ancestral unceded land of the Chechenya-speaking Ohlone people, who are the successors of the sovereign Verona Band of Alameda County. I hope everyone who's in this talk remembers that members of these communities live and work amongst us, and that many of them are our colleagues. Other types of communities, other than humans that you see here, in this audience, you're very well the ones on our bodies. And this is just a stock image, one which I really love the illustration of it, showing that on any surface, you can see many different communities of bacteria. Sometimes they're all of the same clone, sometimes they're different. They have their own neighborhoods and addresses. And yet with all of this microbiome research, we know that there are many different bacteria that are living in these communities. Some are battling with each other. Some seem to just not care who their neighbors are. And some seem to cooperate and crossfeed. We know that the organisms and the phage in these microbiomes influence each other. And yet no single lineage of a bacterium lives in every single microbiome. And specifically in some individual microbiomes, however, you can see a single lineage dominate or a single species. And this is the part where we're going to focus on. And I want to ask the question, so for those individual microbiomes, how does a strain, or in this case, a genetic lineage, build and maintain a community, and particularly when it's in a complex niche? And so the choice of organism that we choose is called Proteus mirabilis, and that's because it is found in at least 40% of humans. It's in your gut. And we know that when you do deep sequencing, you find one lineage in a person. That lineage will then, if you're unfortunate enough to have a long-term indwelling catheter or a disease such as rheumatoid arthritis, travel from your gut to your bladder, where it can cause persistent and recurrent bladder infections. That migration is again caused by the one that's already inside of you, not by your neighbors. How Proteus achieves that is that when it leaves the gut, it's thought to differentiate into something called a Swimmer cell. And so that's seen here, and it's actually the inspiration for its name, Proteus, meaning of the gods who's a shapeshifter, and mirabilis for it being absolutely miraculous. So again, here on this, what you're seeing here is a transmission electron micrograph where we've inverted the colors. Black is the surface. The big rods, the big bodies you see there are the Proteus cells themselves. And those little white legs you see coming off, those are the flagella. So when Proteus, which is usually about 2 microns when in a biofilm or in your gut or in liquid, hits a surface or anything that's highly viscous, it'll differentiate and grow up to 50-fold in size. These monsters have multiple copies of their chromosome, and they move rapidly together in a motion called collective motility. And you're going to see that here in this movie. So before I start it, I wanted to tell you that this is 5x real time. This imaging is using microscopy where you have an air interface. The cells themselves are embossed. About 10% of them are in green fluorescent, are constitutively expressed in green fluorescent protein, simply so you can see who they are. The gray in the back, that's the surface itself. And so these cells are moving across the surface from an area of high density to an area on the northeast corner of low density. And I'll just let you watch it for a minute. Sorry, not a minute, like 15 seconds. So I hope that what you can see is that these Proteus cells, once they differentiate into these large monsters, are able to gather together and quickly and rapidly use collective motion to move across the surface. In fact, at body temperature, as a group, they can go a full centimeter in an hour without any cell division. They move in multiple different directions, whether it's, and they don't really care, and they can turn around in circles, forward, back, a whole thing. The key part, though, is that this migration requires cell-to-cell interactions, and we know that there's communication between them. And this is the basis for how they form what we call a swarm colony. And you'll see that here on this movie. Like this is a 10-centimeter diameter Petri plate where we've inoculated when we were starting the movie, there are about 10 to the 8 cells in the center. I will again let it play. This was taken over the course of 2 days at room temperature, so simply to slow them down. And I hope what you can rapidly see is that as a population, Proteus is able to migrate over centimeter scale distances. When they're doing this migration is also when they're expressing all their own proteins, like ureases. And so the collective behavior, the migration is all correlated with their virulence. And if you actually block them from migrating, block them from entering into this collective stage, you can stop infection in a mouse model. This pulsing you see, where you see this rapid burst right here of cells going out, is when they're in those long periods of having those big swarming bodies, followed by periods when they stop. We do not understand a lot about the development cycle, and that's not for today. But nonetheless, I just want to remind you that outward migration, what you're seeing at that point are non-dividing cells who are swarming against each other, who are actively talking to one another. And you can see that in the black bodies on top here. And when they stop, they divide, they fill in the spaces. And so this is actually a rapid way for them to occupy new spaces and basically prevent other organisms from invading the areas that the swarm has taken. So the last bit of intro I want to give you before I launch into the talk is that one of the reasons we choose Proteus is because we know, since we know that humans have different lineages from each other, we can take a strain from one patient, an infection, in this case of the blue patient, and put it against the strain from the orange patient. And what happens when they're allowed to swarm, not when they're in biofilms, not when they're in liquid, but when they're allowed to swarm, is that they will eventually form a boundary and differentiate between the patient, the lineage from the orange patient and the lineage from the blue patient, which you can see by a line starting to form in the middle of the plate over here. However, the blue versus blue doesn't form. We know that this requires physical contact, and we know this is not something that happens right away. They have to actually merge and interact with each other first. And so to summarize a whole bunch of work in my lab, and it always makes me very sad because a lot of grad students, postdocs, and undergraduates, as well as postbacks, who put their effort into the summary. However, we're going to summarize in 20 seconds. What happens is that when what you can see here in the micrograph on the right, we've pseudo-colored it so that the surface is in purple. Dark cells are populations from one cell line, and yellow are cells from another cell line. When the cells elongate, they start turning on something called the type 6 secretion system, which you can think of as a giant sword. And only when they're in type 6 swarming do they form 10 copies of this, all from a single locus across their body. And they take these swords and they stab their neighbors as they're moving. And they stab indiscriminately. Siblings, foreigner, it doesn't matter. At the top of that tube, of that sword, are sometimes proteins that we call IDS, doesn't matter for now, that don't kill them. And this is a secret signal that we know is only for within strain communication and leads collective behavior. Ignore all of that. Beautiful work, we're just gonna ignore it. Today I'm gonna call it killing, because killing is really kind of cool sometimes. And in this case, we know that we've known for a long time that Proteus includes a little toxin on some of these. And they also stab their neighbors. And I'll be honest, when my grad students, Abby and Denise came to me and said they wanted to study it, I said, really? Oh, there's a lot of types of work on toxins, and it's gorgeous. Is it going to be any different? And they said, we don't know. So I let them go. Luckily, it's a little different. So this is work that's combined from Dr. Abigail Necht, Abby, who's now a postdoc in my lab, finishing up some follow-up work, and Dr. Denise Sirius. So the general sense is that we're going to take this only from the perspective of the cell in the center. As these cells are migrating across each other, the cells are going to stab each other. They send off an effector protein. At that point, we didn't know what it was. It's going to be Pac-Man here. If you're a foreigner, so this is in green, you'll eventually die. If you're the immunity protein, which is going to be in the peg shapes, siblings have them, they bind, you're supposed to survive. This works beautifully, fits all of the dogma, all of the hypotheses as we expected. We know that king bacteria always have the immunity stream downstream of the effector based on our work and other people's. And we also know that these immunity proteins are supposed to be specific. If you have the PEG, you're going to survive. If you don't have the PEG, sucks to be you. So first of all, to get after this, we have to ask, what is the Pac-Man doing? Long story short, this is predates of AlphaFold 2. So we had to look at the protein structure. We now understand that it's a DNase, and this is just the data to show that. This is a DNA gel after we've done an in vitro DNase degradation assay. So we actually purify our effector protein, and we add it to lambda DNA, which you see in the pure DNA column. With a negative control where you don't have enzyme present, all of the lambda DNA is there. This is with methylated lambda. If you add our enzyme in increasing concentrations, you can see that it degrades over time by the loss of the lambda band. And then finally, if we mutate a single site in the active domain of this enzyme, we can actually block degradation. This works from unmethylated DNA as well as plasmid-based DNA. We show in vivo that it does the same thing. So DNases, if you express them in the cytoplasm, you should get rid of DNA. So on the left, you'll see the phase contrast images, where on the black is the cells, gray is the agar surfaces. And on the right column is DAPI simply staining the DNA that so you can see it fluorescently colored. In this case, will be a white. If you use an empty vector, these are proteus swarmer cells. You can see the beautiful cells. You can, if you might be able to tell, but there's like a dot of ants within these long cells. Those are the chromosomes. If you express the protein RDNE, which is the name of the DNase, it's a giant mess. There is some DNA signal still, but these cells are dead, and we've shown that through colony forming units. However, if you add its partner cognate immunity protein RDNI, the cells recover, and we see beautiful swarmer cells and DNA again. So this, in fact, is targeting the DNA, and if you express the RDNI protein, it protects. However, when we do these assays, we're expressing them in the same cell. I want to remind you that one of the cool things about this type of communication is that you have an enzyme that's coming from one cell that's being shoved into the neighboring cell. So it's the neighboring cell that actually has to survive. So to show that, we've developed an assay in the lab where we can look at protein transfer and ask whether you still see survival. And so this is based on that swarming phenotype. Again, so in the center is the mock plate. In purple are the cells that are going to deliver the toxin. That's called BB2000. So they will always be on the top left of your plate when you see these. On the right is the receiving protein cells. Those are protease marble cells from another patient. And in fact, as a side note, this green strain was the only one isolated from the Human Microbiome Project, where you had a non-clinical, where there was no infection associated with it. This guy does not have any of the things necessary to protect themselves. So they're basically sitting there vulnerable. And then they mix them together one-to-one on the bottom of the plate. We know that these factors are what helps to make that boundary between the cells. And so in a control experiment, which is the first thing you see on the left, you can see here's again, that wild type is purple one, on the right is the green, and on the bottom is the mixed population. If you look where the white arrow is, you'll see that there is now a line between the competition and the green strain, as well as a line between the purple and the green. And so below that tells us is that the wild type, the purple, is outcompeting the gray. So for the rest of the talk, rather than having to figure out all the different lines, you can just look at Abby's nice key below. Gray means you win, white means you lose. In populations, when you now add transgenically the RDNI from that binds the RDNE, what we find is now that ATCC strain, now that it can protect itself, it wins. This is what you'd expect. If you add GFP to that ATCC strain, they die. So you have to have the specific shield, the PEG, to survive. We next wanted to make sure that these actually bind in vivo. And so this is shown in two different ways. One, we're doing a bacterial 2 hybrid on the left. The short answer of this is that if you see blue, that means these proteins are binding E. coli. If you see white, they're not. Whether we put the link one of the proteins on either the T18 linker with bacterial 2 hybrid, or we put it on the T25, you'll see that RDNI will bind RDNE, and RDNE binds RDNI. And that's also seen in the pull downs on the far right, where the controls are flagged BAP. It's a protein that's that's an E. coli, but not in Proteus. We use it simply to clean up the gels. And the next ones over are the RDNE flag. The first two sets of columns are an anti-flag showing you that in fact we're making the RDNE protein or the flag back protein in lanes 4 and 8. That's what we're eluting down. And then when you look at the anti-2 strep, you can see that even though there's RDNI in both present in both populations, only the column that has the RDNE flag, do you see the RDNI come down with it. So these two proteins are binding in vivo and in vitro. They are partners, they are cognate. Last thing to show that's an effective immunity pair is that they're found next to each other in the chromosome. And so I'm going to go through this really rapidly because what matters is not so much the details of this. What matters is that in Proteus we find RDNA, which is at the tip of a gene. It's the process C-terminal domain of a traditional RHS factor. It's immediately upstream to the 5' of RDNI, which is in magenta. Abby took this sequence and then looked across all the bacteria genomes that were closed or mostly done, and she was able to find that this doublet of genes is found in a wide array of bacterial species. And they're always closely associated either type 6 or type 4, actually, or other transport systems. But more importantly, when you look at the genes that have the genomes that have this, they're scattered across the bacterial tree, which you see on the far right. They're not clustered just to proteobacteria, other proteuses. They actually found all in the firmicutes. And someone earlier talked about firmicutes and lactobacillus, Maria. You know, it was beautiful. When we think about these effector proteins, we tend to think of them only within one subgroup, especially when we're talking about siblings. So with that, Abby said, wait a second. Why would a firmicute have an immunity protein that can bind to, that looks like the one that you have in Proteus? I should also point out the firmicute that was there, Raphia, is in your mouth, not in your gut. So maybe it's just transfer. So we took a closer look at these and asked how similar are they actually? On the top here is simply using predictions of the secondary structure for these proteins. RDNE is on the right, RDNE is on the left, RDNI is on the right. About 20 of those of our favorite across the entire tree. Red are alpha helices, blue are beta sheets. And then below, we've now color-coded in purple are conserved amino acids, conserved residues, and gray is less conservation. And what we found was that there's basically no sequence conservation, especially when we got to RDNI. They don't look like each other. There, with RDNA, you can really see the structural fold, this PDDEXK motif. You can see, especially when you're doing AlphaFold2 motifs of them that you overlap. You can very nicely see and kind of see how they might be similar. The immunity proteins, honestly, when they said, "Oh, look, these are similar," I was like, kind of. And the conservation especially is maybe there's a region there that seems like it might be conserved. So the first thing we did was then say, okay, can these actually act as similar proteins? And so using that competition assay I mentioned before, the swarm assay, we then, rather than introducing just the one from Proteus, we introduced 3 others from either Raphia, Prevotella, or Pseudomonas. So again, you've seen this assay before. The left is the guy who's shooting the toxin. The right is the one who's receiving it, totally vulnerable, and the mix is below. The 2 plates you see on the first column and second column, this is again what you saw earlier, where the Proteus immunity protein is able to help the ATC phase strain be protected. To our surprise, the immunity protein from Raphia, that gram-positive loop in your mouth, actually protected against the Proteus effector. But that wasn't true for the Prevotella or the Pseudomonas. So then we asked, do they bind each other? I'm just going to show you the bacterial 2 hybrids here. And sure indeed, the Raphia, which is in orange, does bind the Proteus effector. As I said, when we looked at it, the only thing that really seemed to be conserved was this region in the C terminus. So we looked at the conservation of residues within it. We found 7 highly conserved domains. If we mutate them individually, there's absolutely no impact on the protein. We have to mutate all of them together in order to make the immunity protein no longer work. So knowing that, we then asked, is this region sufficient to confer specificity onto a protein? And again, you're going to look at the same competition assays. And now what we're doing is on the first one is the empty vector. The second one is the first one is not empty vector. The first one is just from Proteus. The second one is a Proteus immunity protein, but now with that conserved region from Rothia. Third is that Proteus protein with conserved region from Prevotella. The next one is from conserved region from Pseudomonas. And then we just swapped them out so that now we look at the Prevotella and Pseudomonas, which could not bind before, and ask whether or not the Proteus C-terminal region is sufficient. And what you can see is the only ones where you see protection are when we have either the Rothia or the Prevotella C-terminal domain in the Proteus backbone for that immunity protein, indicating that yes, that C-terminal domain is sufficient. Can provide some protection, but it's not universal. Surprisingly, we found though that even though the blue-purple hybrid here doesn't protect in our in vivo assay, it still binds. So simply binding up the immune effector isn't good enough. There's actually another step that's happening. We don't know what that is. Before you ask, we don't know. We are searching. Okay, so just quickly in conclusion here, this is great. I'm a molecular biologist and a geneticist, so I love having systems that we can do in the lab. One of the questions we had though was that if this is happening truly in nature, we should be able to find, would hope to find or expect to find some signal in actual human microbiomes of this. I don't like working with vertebrates. There's not a single mouse in my lab. However, if you'd like to look at your mouse studies, let me know, because I would happily let you look at it. So instead, we took the easy way and looked at all the published metagenomes for humans. And this was in collaboration with Dr. Daniel Utter, who's now a postdoc with Victoria Orphan at Caltech. And this was when he was a graduate student with Colleen Cavanaugh at Harvard. And what Dan did was that he took the sequences, the DNA sequences of RDNE through RDNI for Proteus, Rothia, Prevotella, or Pseudomonas. And he then ran it across every metagenome that we could find in NCBI. We tossed out everything that wasn't human metagenomes because we actually didn't, except for, that's not true, I take that. The data I'll show you is from the primarily human metagenomes. We did not find it that much in terrestrial for the 4. What we can then do is that then he can map the reads, the raw reads from the metagenomes onto our sequence and ask, do you see evidence of RDNE, RDNI, or both of them? And this is critical to understand that this data is only for E&I pairs that are in a single uprun. We're not looking at orphans in this data set that I'm telling you. And what we saw was that when we looked for the coverage of the RDNE in all of these metagenomes, we see that in the human oral microbiomes, we see much evidence of Rothia and Prevotella, as you'd expect, since they are human ones. In the human gut, we mainly see Proteus. You do see some Rothia and Prevotella. And in the terrestrial ones, there's not really much there. Pseudomonas, that's where it'd be. We don't see Pseudomonas, Proteus, I can tell you why that is. However, the key part is that if we look for where do we see in a single microbiome the co-occurrence of more than one of these 4 together, we saw that in only a subset of them. And what we've done here is map each of the distributions. And you can see there was one person who was unfortunate enough to have Prevotella and Proteus in their gut. And then there's a much bigger overlap between Prevotella and Rothia. And we're starting to explore that now in the lab. So let me just finish up and tell you what this means. In the traditional model of effector-mediated pairs, especially when you talk about type 6 secretion, the idea was that it was a one-to-one interaction. Your immunity proteins only for survival with your sibling. Our data suggests that that's not true. These immunity proteins, fundamentally, they are proteins. If they bind something and they can still function, they're going to do that. And so if that immunity protein in a cell sees an effector from somebody who's different, they might survive. So that has to change how we think about microbiome structure. That's not just you can only see your sibling. You can get further protection. And I'm not an evolutionary biologist, but I think that there's some questions as to how this might be selected for over time. We also now know that it's a 2-step process rather than this 1 step. There's clearly a step after binding. We don't know what that is. Finally, we've been really, this article just came out this summer. We're super excited because in the last 6 months, several other articles have actually come out and shown in vitro cross-binding between effector and immunity proteins, though they have not followed them up yet with in vivo studies. And most of those are orphan proteins. Okay, so with that, I want to thank the folks who are currently in my lab Abby is the one to the back behind the person in the yellow shirt. She is on the job market for another postdoctoral fellowship or job if people are interested, a fantastic biochemist and illustrator, as well as the funding sources for this work. And a lot of the protein work was done in collaboration with a lot of advice from Rachel Godet and her lab. And I can answer any questions.
That was a really great thing. I was just curious if there's a cost to producing the immunity proteins, because you showed this ATCC strain lacking a lot of these, and it almost looked like it was growing, it was overtaking the plate in some experiments.
Yeah. Good question. So you're asking about what are the costs of making these proteins? What are costs of? We don't. The short answer is I don't know, but I can speculate about a couple of things. One of the reasons that this works and why we like looking at the social bacterium is that because they're moving across the surface, we can actually see what's happening in the cells as they're going along. The ATC strain, for reasons that are not known, actually moves much faster than the wild type strain than the clinical isolate that we use. And so if you're not, for example, if you get rid of all of the type 6 in our strain, the ATCC strain competes, like it'll just take over the surface more quickly. We have not done any metabolics and associated with the type 6. There's some reports that I've seen suggesting that the type 6 doesn't actually have that big of a metabolic cost on cells. All of that's still pretty much early days, and they're of course not done in Proteus. They tend to be done in Pseudomonas and coli, better strains. I don't know.
Yeah, great talk. I was curious, like, what's known about the host range of type 6? Can it actually cross really distant species? Or?
Yeah, so in Vibrio cholera, which is where much of that work is done, it will, infect amoeba, and also, so it'll cross into eukaryotes. It's essentially, the thing I like thinking about the type 6, again, I'm a molecular biologist, and so I tend to think about how do proteins work and how they function. This thing is basically a phage tail that jams into anything you get into. And so I think the question is, which host ranges have we not looked at? Right. We don't know who Proteus is talking to in the cells. And one of the things I would love to, so I don't know if you saw, but it was really Prevotella and Raffia who had the overlap. That's where we want to go next. We want to understand for these guys who really do have to find spaces in the oral microbiome, do we see this type of correlation between it?
Karen, just following up on Peter's question, so you have not tried any competition in liquid cultures between, for example, gram-positives and gram-negatives to show whether these effector proteins can kill other?
Yeah, so with Proteus, we've done all, I'm laughing because the number of reviews we've done, answers to reviews. Proteus does not make the type 6 when it's in liquid, or at least, sorry, the Proteus strains that we study, so this one, HI, et cetera, we do not see the genes turned on in liquid or in biofilms. And so we do the competitions and you can see differences due to other competitive factors like CDI, but you can't necessarily, we don't see them based on these genetic factors, which you can, you have mutants in them, we can show them operate. In other species, most definitely, right? You see it's really beautiful in those. We have done competitions with Proteus and other bacteria on surfaces, and there we can see that the type 6 is relevant, and particularly the Ardennes group that's telling about. Okay, thank you.
Okay, thanks so much to all the speakers in the morning. It was really exciting work, and we're gonna break now for lunch. Please come back around 1 to see the posters out in the lobby.
Yeah, so we're gonna push posters to 1:15 just so everybody has a chance for lunch. So lunch is right outside these doors. You're welcome to eat outside through the glass doors above Ladle Leaf, and we've also have additional seating up the stairs in the nursing mezzanine. So find a spot to eat, and then at 1:15, kind of come back to the lobby for the poster session. Thank you.